Hello, hello, hello. Welcome, everybody. Welcome to the stream. Hello, hello, one and all. Welcome to our Valentine's Day super special, where we will be talking about cell polarity. So I framed this stream as a stream talking about fertilization. Fertilization will come up in this stream, but only as a player in a topic that I am much more fascinated by, and that is, how does a cell understand its head from its tail? But before I do that, let me begin by saying hi to you all. Hello, Robert. Hello, Clockwork. Welcome to the stream. Clockwork, if you're watching my channel and you're not watching his, I don't know what you're doing, but Clockwork recently released an epic video. And epic, I mean epic as in like how you should use the word epic, as in it is 40 plus minutes long, about eukaryotic transcription, which I highly recommend you go watch. I will be watching it today, and I've actually played with the idea, and Clockwork, you can feel free to like tell me no or whatever, but like going forward, maybe we do like unarchived watch-alongs. I think that would be really fun as a community, so that you can hear my live reaction to the wonderful work that Clockwork does, because I just think that would be fun. It's kind of a shame that I watch them alone. So I just had that idea like 2 minutes before stream, but we could talk about whether or not you want to do that. I think that'd be a lot of fun. Hello, Tangia. Hello, hello. Welcome, welcome. Hello, Dreamless Sleep and Azure Legends. Hello, Nicholas and Jtaro. CR Smith, welcome, welcome. Kevin, hello, hello. Archie, welcome. Flying Taco, welcome, welcome. Masako, welcome. Azato, hello. Freak, hello, hello. CR Smith, welcome. I think we are all set. I'm sorry, yeah, the bot. Okay, yeah, the bot's in. I don't know why the bot takes a couple of minutes just to warm up, I guess. Yeah. So today we are talking about embryology. I just also realized that the Stream Elements bot did not paste in the link to the award. So if you would like to nominate me for best lore for the small VTuber awards, let me just get a real quick link in the comments. It'd be kind of cool to oh man, hold on, sorry. I need to grab this from the thing, copy this, paste it into chat. Here we go. Hello, Jabu. Hello, hello. Yeah, so that is, I'll actually pin this. Vote for me in the small VTuber awards. It'd be really interesting to see, even if we don't win, how much of a niche we have carved out in the VTuber space, because I don't really expect to win per se, but I think it would be fun to see collectively, like, this idea of, is there a space for science communication VTubers, and seeing the statistics, and if we get shortlisted, it's a very good indication that we do have some weight behind us and that we are growing. And even if we don't get shortlisted, it's still an interesting check to see how far we've come. So I think that'd be pretty fun. All right. Truly the question of ages. How is Babby formed? Exactly. Welcome in, Marlon. Welcome, welcome. Hello, Pedro. Welcome, welcome. Okay. Oh, okay. So I've been very, very excited to stream this particular topic all week, because I realized that Valentine's Day gave me the excuse to talk about one of my favorite topics, which is cell polarity. Favorite not in the way that I am an expert in this, because as I've said on this channel multiple times, developmental biology is like the final boss of biology. It is so complicated with so many moving parts, and you have to understand everything about the cell and about chemistry. Not everything about chemistry, but everything about biochemistry. Like, developmental biology exists to test everything you've learned in your biology education up until the point you take dev bio. That's why you learn dev bio last, right? So today is not going to be a comprehensive lecture on developmental biology. I need way more time to do that. Instead, we are going to tease an interest into the field of developmental biology by taking a look at one core idea behind how organisms form and how cells talk to each other. So this is going to focus mainly on how and when does a growing embryo recognize its parts? And that is a surprisingly deep and complex question that often gets ignored in a lot of biology classes up until you get to, well, I learned about this in graduate school. I mean, maybe nowadays kids are learning about this stuff earlier, but for me, it was always sperm enters egg and then a whole bunch of stuff happens, but no actual explanation of why or how they happen. And that's what we are going to get into today. Developmental biology is more than just the study of the growing fetus, but it also has broad applications in cancer, fertility, reproduction, infant health, prenatal health, and of course maternal health, right? So this is a very, very important field of biology, and I am very happy to be able to start talking about it as opposed to waiting until I've provided enough of the background information. Because if you've watched my content before, or if you've taken biology classes, you should be able to understand up until this point. You told me on the Discord to ask on here how gametes roll back senescence-based DNA damage like telomeres. I have not gotten to that, unfortunately. I did not have the time to look into that particular question. But when we talk about longevity, so longevity and dev bio are related, but they are unique enough in the way that I will have to do a longevity 101 and a dev bio 101. So they are related, but not the same. So C.R. Smith, I'll definitely get there downstream in my 101 series. All right, but without further ado, let's get right into it. Okay, so this is the ultimate question, right? How does the embryo actually know its head from its arse, right? And so if we take a look at this very simple schematic where I have a zygote or an oocyte, in mammals, this cell goes under a series of cleavages, so the cytoplasmic volume doesn't change, but the cell divides nonetheless. And at some point, these four seemingly identical cells become, a person with polarity, right? So there's an anterior end where the head is, a ventral end where the chest is, the dorsal end where the back is, and then the posterior end where the butt is, and a whole bunch of other different terms to describe the different axes of the body. But it's not entirely clear, at least for a lot of your biology education, how you go from seemingly identical cells to a thing that has patterns involved and polarity. And what do I mean by polarity? I'll get to that in a sec, but before I figured it might just be good to watch this old ancient Phi short, about when I first talked about anything related to developmental biology. Hello, welcome in. So this is a short that I did way at the beginning of my channel, talking about dev bio, the first time I've mentioned it on this channel. So let's go ahead and play that. Okay, so you want to be cradled by the many strong, comforting arms of your zaddy Machamp, but there's just one issue. Machamp doesn't actually exist, but could he? See, if you want to turn your Machamp into a Machamp, you're going to need extra arms. And to get those, you should know about Hox genes, a set of genetic codes that control where and how your limbs end up developing. A lot of this work was done in fruit fly mutants, where the Hox genes have actually been mapped out to the entire fly body plan. See those antennae? They're actually legs. So thanks to our understanding of the Hox genes, you may be one step closer to your ultimate fantasy. I like this. I like this short a lot. Prehistoric Phi content. It's just regular history Phi content. So the Hox genes are genes that are commonly referred to when talking about how the different parts of the body form, and how you know where to put a leg, how you know where to put an antenna. But there's a sort of chicken and egg question in this. Oh, okay. There's a kind of chicken and egg question where, okay, so the Hox genes determine where, your arms go, where your legs go, but how do the Hox genes know where to appear, right? So the Hox genes you can think of as sort of programs that are activated locally in certain segments of the body. And so where you get that program turned on, you get an arm or a leg, depending on which program is active in that location. But if we peel back the timeline just a little bit, what sets up those Hox genes, right? We're talking about kind of how I'm romanticizing as like the Big Bang of life in a kind of way, where I want to take a look at some of the first cues that allow a developing embryo to even determine where to put those Hox gene programs, right? So here is one of the most central themes in cell and developmental biology, is this concept of directionality and polarity. So a perfectly symmetrical cell has no idea what directions are, right? So imagine you're a perfect sphere and you're floating around in the cytoplasm. You don't really have a physical way to determine what left, what right, what up and down are. And that is distressing, right? So if you are perfectly symmetrical with no polarity, and that's what we're referring to as polarity here, right? The ability to have poles on north, south, east, and west. But if you're a perfect sphere floating in liquid, you don't really have an idea of what these directions are. They could be anything, right? And so we go back to this question of how does an embryo know its head from its arse? And a perfect sphere doesn't really have a good idea of where the directions are. Don't they have gravity to determine up and down? Not all organisms rely on gravity for up and down. If you are suspended in liquid, the effect of gravity might not be strong enough to actually make a difference, depending. But that's an excellent question. I think, and I don't want to get it wrong, because there are so many, when you learn about developmental biology as a molecular biologist, you have to learn how so many different embryos develop. I'm only focusing on 3 here, but I think, and I could be wrong, and I don't want to leave people with a wrong impression, but I think that is true for frogs, for xenopus, but I could be wrong on that. So gravity can have an effect, but gravity might not have an effect for every organism involved. But you're thinking very, very correctly. Thinking about cues, thinking about ways that we can break symmetry, things that are going to affect a cell differently depending on its position, and gravity can be one of the factors. Okay. So we have here an image of a developing C. elegans worm. And so I want to point out that even if cells look the same, right? Like this is all just a ball of cells. Eventually you get right here at this stage, it's the, oh yeah, my cursor is on the screen, great. Here you get this sensation that there is something that is broken here, whereas everything up until that point looks pretty even. So at some point, even if cells look the same by eye, they have to differ in some way in order to give rise to asymmetric complexity. And I think that's very, very cool. And so what we'll actually realize is that even though all of these cells look the same, they're actually immediately different. These 2 cells you can already see are different, right? One cell is a little bigger, it pushes into this cell just a little bit. This isn't just luck, this is a very characteristic representation of how the C. elegans worm develops, right? Big spherical sphere, and then this one that's kind of concave in. So we can tell in C. elegans, in the worm, that polarity is established pretty much immediately, which is very, very interesting. Okay. So, the start of another precious life, isn't it? If you, I think there's a lot of romanticism in developmental biology where I think that because the process of like quote unquote hacking the formation of life, not hacking, that's kind of rude, not hacking, but rather, you know, the way that I like to think about how to tell stories in science is by providing interesting questions for the audience to think about. And I think one of the hardest questions to even ponder is how do you go from 2 cells into a complete complex organism. To me, the way that cells do that is incredibly complex and just an incredible result of evolution, which is very, very fun. I know it's true for seeds. They done it with seeds in a centrifuge, and roots grew towards the center while the body grew towards the edge, but that's a different kingdom than us. Yeah, I know very little about how plants develop, but that is interesting. That is interesting. Is there also going to be anything said for how the embryo of many species look very similar at a phylotypic stage? I will touch upon it. There's actually one slide. On that idea. Okay. So I kind of introduced this idea of cell polarity. So let me define it here. Cell polarity is an intrinsic asymmetry within the contents of the cell, right? So cell with no polarity, this is something that has no edge or no directionality. It's just a sphere floating in space. And then cells with high polarity are something like neurons, right? Where you can clearly tell that there is a head up here and a tail down here. So we can already tell that there is some level of difference. And so this is a really, really important concept to grasp, that this is what I mean by polarity, right? Differences across an axis of a cell. And so you need some sort of polarity in order to establish your axes. So again, at some point that symmetry is broken and cell polarity is established. So even though we have cells that look similar, maybe these two are getting something different, and maybe these two are getting something different, and that's maybe how you tell your head from your butt. And so the way that different organisms do this is actually quite different, which I think is very, very interesting. Okay, so while the gross anatomy of embryos of many animals might look similar, the underlying processes are surprisingly different, right? So this is sort of what Marlon was referring to, the fact that if you just look at the developmental stages of different animals, so we want to look up and down here because these are different species of animal, we can actually see that there are a lot of common similarities as to how animals develop. And that's certainly true, right? Like the process by which we form animals across related genuses is certainly very similar, but the beginning of it is actually surprisingly different depending on what animal you're talking about. Certainly if you're talking about mammals, there are going to be more commonalities between how mammals develop than how chickens develop, than how flies develop, than how worms develop, right? So even though this is a common idea to check for what am I trying to say? Even though the similarities between organisms in their embryonic development is an indication that these processes are well conserved, if you go out far enough in the animal kingdom, you actually find that animals have all sorts of crazy strategies to start this process. Even though by this stage, by the time you get something that looks like an animal, things from here tend to progress pretty similarly. Okay, so here's the first instance of where I want my audience or my viewers to really ponder how you would hack this. So imagine a completely non-polar, uniform, homogeneous egg. What's the simplest way an embryo could gain polarity? And so I know that there have been comments in the chat suggesting gravity. And again, gravity, I believe, is correct for certain species. Someone had mentioned that that's true for some plants. And it might be true for xenopus frogs, but I don't fully remember how frogs develop. But what's another simple way an embryo can get polarity? All those examples are chordata from what I can tell. That's true. I think that's probably why we look at those particular model organisms for embryonic development, is because there are chordates. I am not like an animal biologist, so I can't remember exactly where to draw that line, but we are going to be taking a look at three very different animals and how they develop today. So perfectly symmetrical egg. What's the easiest way an embryo can gain polarity? The placenta is not there yet. That is correct. We're not talking about the placenta at all. We are just talking about a purely hypothetical example. No need to think about the placenta, although the placenta can be something interesting to think about, right? Yeah, a disturbance of some kind. Yeah, but what is the simplest disturbance an egg can experience so that an embryo can develop? This is a kind of deceptively hard question. You're gonna have to think a little bit about how an embryo actually develops. And it looks like we have some good answers here. So Silas and Ari say sperm entry point. C.R. Smith says, could we use the sperm enters the egg? Exactly. Yes, exactly. So one of the simplest ways an embryo could gain polarity is sperm entry. So very, very good. Very, very good. Awesome. So yeah, so I see other examples in the chat which can be symmetry breaking events, but I wanted to start off with a bit of a something that we might all be very familiar with. Yeah. So the incoming sperm, right? So this second by which this perfectly spherical egg touches this sperm, that is a potential symmetry breaking moment, right? So even though this cell has no intrinsic polarity, it has no idea what left, right, up, and down are, the second it touches a sperm, it all of a sudden knows, okay, there's the sperm side and then the non-sperm side. Bang. That's what we're talking about when we're talking about symmetry breaking, when we're talking about cell polarity, that moment of determining directionality. All of us have experienced this, truly universal experience. Exactly. Okay, so this model of symmetry breaking is actually true for C. elegans. So we actually looked at developing C. elegans earlier in the slides. So the way that an egg from the worm, the C. elegans worm, knows its head from its tail is this super simple symmetry breaking event where the sperm enters the egg. Okay. So conceptually, this makes sense, but I wanted to give you a little bit more of a molecular view of what about that sperm entry sets off the development of the embryo. Okay. So there is a class of proteins called the PAR proteins, which stands for partition deficient proteins. A lot of the names of proteins comes from the fact that in classic molecular biology, you do like random mutations to animals, and then you check for ways that they differ from your non-mutated animal. And when you do this mutation, what you can find is a set of C. elegans worms that never develop normally, that they are arrested in whatever stage of embryonic development. And the genes that have been identified from those screens are named after the phenotype, aka how the animal looks. So a lot of these proteins are identified as partition deficient proteins, as in they do not do development correctly. And so the entry point for these sperms sets off a series of molecular cascades that partitions these proteins. So let me explain what we're looking at here. We're looking at the maternal egg and the paternal sperm. After the sperm enters this egg, you have a partitioning of the red proteins, PAR 3 and 6, and the orange proteins, PAR 2, and it should also be PAR 1 at some point. And so there's something about the sperm entering the egg that causes the red proteins to get pushed back by the orange proteins, right? So all the orange proteins that are happily floating around in the cytosol of the egg, after that sperm enters that egg, that cytosolic protein ends up on one side of the membrane, specifically where that sperm is. Okay, so how does that work? So at the entry point of the sperm, there is a molecular cascade that causes cortical flow. So some people have already said that, you know, the donation of the centriole from the sperm is what causes this. So yes, so the sperm dumping its contents into the egg provides an organizing center for molecular cascades to begin the ruffling of the cortex here. So as soon as that sperm enters that egg, you can imagine sort of the membrane ruffling, and this ruffling is going to cause the insides of the cell to rotate. So there's flow that goes in, back up, and around, and this gradually pushes the orange protein back up and around towards the back here. And that donation point, that sperm entry point, provides sort of a hook provided by the sperm. So these cytoplasmic proteins are flowing cortically up to the left in this image, up and over, and they are getting hooked onto the membrane. So eventually, after enough cortical flow, you get all of that cytoplasmic protein to hook onto one end, the end that the sperm had entered. So that cortical flow allows us to establish both an anterior end and a posterior end. So we already have one of our axes formed. So we're going to go back and look at chat. Doesn't a sperm break the cell wall to get inside? Yeah, so the sperm definitely has to penetrate the egg. The mechanisms of which I'm not like super well read on. Although someone in chat can help can help me fill that in. The thing about biology is that there's so much of it. There's so much of it to keep track of. Okay. So This is a figure from this review in PLOS One, which explains this quite well. It's a little smaller, so I wanted to recreate it earlier. But so when you have that sperm entry point, what you see is that first symmetry breaking event right when that sperm enters. And then when you have that sperm entry point, you get that ruffling, that cortical flow that moves the PAR2 protein from the cytoplasm onto the hook of the membrane provided by the sperm. And then you actually have mutual inhibition of the PAR6 and PAR3 proteins with PAR2 and PAR1. So not only do you get this hook attachment where the PAR2 protein is attaching to one end of the embryo, there is mutual inhibition where these proteins are inhibiting the presence of each other. So PAR2 and PAR1 sort of push out PAR6 and vice versa, right? So there is a natural border that happens, a natural equilibrium that happens. And this equilibrium, you can already tell in this diagram, is asymmetric, right? So there is some natural breakpoint by which there is an equilibrium established by these two sets of proteins inhibiting one another. And because there is that dividing line, that there is this natural equilibrium, that is the axis by which you get that split. And so when we saw those C. elegans embryos earlier in the presentation, we had already seen that there's some level of asymmetry there. And so even at the 2-cell stage, you already get a cell that is slightly bigger and a cell that is slightly smaller. And I think that is very, 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 very cool. So here's my pitch again. I mean, nobody watching my channel should be surprised when I advocate for basic science. By studying the humble worm, we've gained deep insight into how we go from egg to organism, right? Like this idea of a perfectly spherical, infinite potential stem cell becoming a fully fledged differentiated organism, it has to start from somewhere. And by studying basic animals like the C. elegans worm, we can identify proteins that play a major role in the formation of a real embryo, right? So the PAR proteins, the partition proteins are pretty conserved across all animals, right? So this sets up a molecular toolbox by which many, many animals can take from to establish their embryonic axes. Although the process by which that happens is very different, we all sort of use similar tools. And I think that is important to keep in mind, that studying things like worms is really important for human health because worms are so easy to manipulate. One other plug that I want to make for C. elegans research, for those of you who are budding biologists, is that the C. elegans system is 100% genetically tractable in the way that at every single stage, at every single image here, we know exactly what the identity of these cells are. You can trace every single cell from every single stage and know exactly what cell this is going to become. So just by looking at this, we know every single cell that this becomes. And even if we go down like to this level, every single one of these unique cells, we know exactly what cells these are going to become down the line. Like it's absolutely insane. There's a lot of insight that can be gathered about basic biology from studying the worm. And also, C. elegans is also a fantastic organism to study longevity too. So if you want to take a crack at extending human life, you might want to look at C. elegans first, just because there's a lot to learn from there. All right. Okay. So let's actually take a look at a video on the development of oh no, okay, give me 1 second to actually download this video. I actually made this in Google Slides before I downloaded it, and if you do that, then you don't actually get that working. So let me put this into the PowerPoint real quick, because I like to do it in PowerPoint when I'm back at home. But in the meantime, I want to kind of hear chat's thoughts about what you think about what I've covered so far, your reaction to learning about how symmetry is broken in the worm. While I get this set up, I want to hear your reactions. Okay. Okay, and I'll read those reactions after we watch this video. Okay, so let's have a look here. So that's the sperm nucleus and that's the egg nucleus. They merge, and then we will have that first division. We can already see this asymmetry, bigger cell. This is the anterior end, and this is the posterior end, right? And then we have that really, really fine dividing line. And then we actually get asynchronous divisions, right? So the anterior end divides first, and then we have the posterior end dividing second, and that always happens in a normal dividing cell. I think this is a pretty mesmerizing image to look at. So we get that merging of the nuclei, we get that differential partition, we get asynchronous divisions of the anterior cells coming up. Very, very cool. I think it's super cool that you get that asymmetry right at the beginning. Is that fluorescence? Yes, yes, yes. It is fluorescent labeling of the nuclei and these membrane proteins. Yeah. So if it's the sperm that creates the asymmetry, then what about organisms that reproduce by parthenogenesis? Again, development is like super crazy complicated. And I am nowhere near well equipped to talk about parthenogenesis. Just not trained in that field. Since one of my lab mates did all our work on C. elegans, reminded me of our lab meetings. Yeah, C. elegans is fantastic. I really, I don't know nearly as much about C. elegans as I do other model organisms, but I do remember really, really enjoying learning about the worm back in graduate school. I mean, stem cell school. Where did the sperm enter? That's on the right hand side, right? So let me play that again. It starts after fertilization, so let me just pause it here. So this is where the sperm would have entered, and that sperm donated its nuclei. This is the egg nuclei. They are going to merge, and then we are going to see division from there. Yeah, last time we watched this video, just because it's such a nice video. Wait, how do worms even get sperm into other members of the species? That I don't really know. As a cell and molecular biologist, I actually know very little about how the animal functions. I'm mostly a how do their cells function kind of guy. All right, so that's C. elegans. Lovely, lovely model organism. So in terms of brainstorming ways that you can have polarity, entry of the sperm can serve as a potential symmetry breaking moment. But as we move forward, we'll realize that not all organisms have their symmetry breaking moment determined by the sperm entering the egg, although this is the easiest way you can think of that. So what's another simple way for an embryo to be polarized other than a cue from the sperm, other than gravity? So I want another set of ideas. And this answer, I believe, is going to be very, very tricky. So while I think it's very realistic that you come up with a hypothesis that matches the C. elegans model system, right? Sperm entering egg, that's a symmetry breaking cue. The next organism that we're talking about does not, or rather it does use the sperm, obviously it has to use the sperm, but its symmetry is broken by something else, not by the sperm, but by something else. Brownian motion, that's a very good thought. So in terms of addressing Brownian motion and how that could break symmetry, Alan Turing, the famous computer scientist, actually came up with a model by which cell fates were determined through a sort of probabilistic lens. And so Brownian motion comes into play when determining cell fate, just not for this particular answer that I'm looking for. And like, so I've seen students complain about this technique to me before, and I've mentioned it on stream a couple times. The purpose of me asking this like trick question is not to like have a I got you like a moment, because I don't think anybody will get it. It's to make you work to the point where when you see the answer, you are going to be more appreciative of how clever evolution can be. Not to be like, not to make you feel dumb, but to make the biology feel smarter. Environmental pH. How would that polarize the cell? Because it has to affect one end of a cell more than it does the other. A very hairy trigger concentration trigger in a noisy environment. That's kind of the idea that Alan Turing was getting into, sort of morphogen concentration gradient, probabilistic determination of cell differentiation. But it's actually a little more straightforward in this this next organism. Maybe some sort of radiation. So it would have to be, I mean, you can use radiation to break symmetry artificially, but this has to be something that an organism can do. So the Socratic method. Yeah, well, to me, when I use the Socratic method, it's to determine what my student does and does not know. And I often explain that to them, that the purpose of me being annoying with questions is not to be like, I'm going to make you feel dumb or put you on the spot. It's to be like, I need to troubleshoot where you don't understand things. So the Socratic interview is for that. I don't know biology, so not knowing biology can make me feel dumb. Fair, true. Maybe epigenetics. So epigenetics can certainly play a role in cell-fate determination, but I'm looking for something that affects a cell in a different way. So something like sperm entering one end. So whatever your answer is, it has to be ended, right? It has to have a directionality. Cell-to-cell junctions. So that is going to come into play as a mechanism for sure. But not the organism that I'm thinking of next. But keep that in mind because that is the correct answer for something down the line. Yeah. Okay, so let's get into the answer here. So this is, again, I wasn't expecting anybody to get this because this is a trick answer. A simple way for an embryo to be polarized is to actually just pre-pattern the oocyte. mRNA of mom. That is correct, Dreamless Sleep. Yes, that is correct. And I think you had mentioned that you were studying for an embryology exam, which is maybe why you already know the answer here. So instead of looking at the egg, you could just build, instead of the embryo getting polarized, sorry, you could just build the egg already polarized. So that is one method by which you can set up an embryo to be polarized, is just make the egg not the perfect sphere. So that, again, that's a trick question. It's a trick answer. I wasn't expecting anybody to get it, but this is also sort of set up the egg already polarized. And so we're going to take a look at how this organism does that. So this is true of Drosophila, the housefly, right? So instead of waiting for the sperm to come in to break symmetry, the Drosophila says, I'm just going to break symmetry immediately by already making the egg with poles. You can make an egg-shaped egg, genius. Exactly, Pedro, exactly. And, you know, it's often the most simple answers that are the most fun because there's this like eureka moment, because nature is quite efficient in the answers it comes up with. What level do you teach? Because dang, this seems so much higher than high school. I teach at the university level, but I try to do so at a level that is interesting and engaging for a broad audience, which is why I like to implant these questions, not as an expectation that you learn everything, but just as a way for you to appreciate how clever, how interesting evolution can be. So if you didn't learn any of this, that's actually totally fine, because high school learning is just about learning things, but university learning is about learning ideas. And that's kind of what is the term, not elitist, but it's kind of hoity-toity to say that, oh, and when you get to, you know, university, you're learning these grand old ideas, but in high school all you're doing is memorizing. But it kind of is true, just, you know, due to, at least in the US, that's how our system is built. You are memorizing things in high school, but in university you're actually trying to understand things. And so in biology, that's a lot harder to do if you're not already familiar with everything you learned in high school. So I'm not exactly crapping on the idea of memorizing in biology in high school, because you have to be passingly familiar with how things work before you get to the fun things. All right. Audio is cutting in and out. Okay. Let's see if I can fix that real quick. So I tried to sort of up my latency a little bit to have the quality better, but that might be affecting the audio real quick. So let me see if I can change something on OBS to do that. Maybe not. I'm intermittently swearing. I'm just that passionate. So I will take a look at what I can do about that probably after stream, but for now I think we are just going to chug along. Okay. Yeah, so pre-patterning the oocyte. So making sure that the egg is already egg-shaped, that it already has bias built into it. That's one way that organisms can develop. So how does the egg get polarized in the Drosophila itself? Well, The way that a fly develops is through something called an egg chamber. So the Drosophila has a part in its body, um, in its body. I'm not a fly expert either. Are there any fly people in chat that can double check me on this? I don't remember when. No. I'm like trying to think about when this actually happens on the animal scale. In any case, what is true, what is absolutely true is that the egg developed in something called an egg chamber, and then in this egg chamber there are a bunch of helper cells called nurse cells and border cells and stuff like that that help the egg develop. So there's a sidedness to this already, right? Because the cell that is destined to become the oocyte, to become the egg, already has polarity. It's getting different information from the cells that border it from the cells that don't border it, right? So already there's a polarity because there are nerve cells on one end and not on the other. So already you have this idea of being able to set up an intrinsic polarity already here. So this is complicated enough for me to not want to get too deep into this, just because Drosophila development will be its own stream, because we know so much about how the body develops from studying Drosophila. So I'm not going to get too deep into it. However, what I will say, and this is sort of what Dreamless Sleep was referring to, is maternal deposition of mRNA along the polarized oocyte sets up the body axis. So part of what's happening here as the egg is developing in this chamber is that the cells of the mom are depositing mRNA, specific mRNAs, along the body plan of the egg before the sperm is even involved. And so these different gradients of these mRNA molecules, is what sets up these various body plan axes, right? So you can imagine at the anterior end, the mom is depositing a high level of bicoid, which drops off. Nanos is deposited at the posterior. The way that these mRNAs are deposited, I will go over in a Drosophila specific stream. It's way too much to cover in one stream. Not too much for you to learn, but too much for me to prepare. Just being honest about this being a part-time fun thing for me to do. I did not have time to get real knee deep into how Drosophila do this. But the idea is that mom already set up the mRNA in the correct locations to have that body plan. So in the egg, this is what these concentration gradients look like. And then what most people who are studying biology at interuniversity level will be familiar with is this setup of gap genes. So these gap genes, and if you study biology, you're not familiar with this, it might be that you haven't gotten to it yet, but if you are studying development, you will be hearing about these particular genes. And these particular genes that have been sort of pre-planned with this maternal deposition of mRNA, after you get more of that development, you get these very striking bands where you have different genes that are turned on by this maternal deposition of mRNA. So in the early embryo, you get these specific lines. And if you've ever wondered why, worms have segments, why insects have segments, like clear, defined body units, there's a reason why we study them to understand our body plan, because our body plan is a lot less obvious that we have segments just looking at like the naked human body. But you look at a naked insect and you can already see segments being formed. And part of the determination of those segments is the expression of these gap genes, these beautiful, defined, hard-edged bands of gene expression that then become different segments later on. So this eventually sets up even skipped and ingrailed genes. And I don't really remember enough about Drosophila development to connect the dots, but I want to set up this as a point of interest for you to pay attention to this channel when I do get to development, that understanding the body plan, we got to look at Drosophila, and then we can make some interesting connections to mammals later on. Okay, so let me go back and look at chat real quick. I really need to relearn biology, the introduction to biology. Biology is something that you can relearn endlessly and pull out more and more interesting things. Professor Dave Explains has nice videos about biology. I'm sure he does. I've seen Professor Dave being recommended a lot on this channel. And development is something that you often have to watch videos for because the processes are so complex. But the thing about copyright law is that I can't show them on the channel, or most of the videos I can't show on the channel. It's interesting to see non-translation related applications of RNA in the cell. Does this mRNA get translated here? Good question. I was wondering that too. I don't want to say too much about Drosophila right now. And so Dreamless Sleep, who's about to have a test, probably knows more about how this works than I do. So you came here hoping this would be helpful for you. It's probably not. You probably know more because you were actively studying this topic than me, who, because the last time I studied this was like upwards of 6 years ago. So I'm a little rusty. So I'm glad to have Dreamless Sleep in the comments, picking up where I have gaps. Yeah. Only flies. True. Okay. So by studying the humble fly, we can learn about how organisms pattern themselves, right? So the different patterns and body segments. And what I do remember about learning development in the Drosophila is that if you do have a wrong number of bands, you can duplicate like a thorax or an abdomen or something, which I think is really, really interesting. That developmental biology is both really complex but also surprisingly simple. Like, oh, if you just have one more band, you get another thorax. Something like that is actually really, really pleasing, I guess you could say, because it takes a good chunk of the mystery out of how we develop. And I think part of the joy of learning biology is having the mystery of life being peeled back one layer at a time. But biology is kind of like an infinite onion where you keep on pulling back layers and you always think you're getting closer to the core. In reality, you have no idea how much there is left to learn. Okay. I think I'm probably gonna have to download this video too, judging from how. Yep. Okay, so a little more scuff. Please hold while I download this video as well so that I can put it on the PowerPoint. But this is a very, very famous video about how the Drosophila embryo develops, and this video is the quality on this video is insane, just like the quality of that fluorescence. So I'll pull it on the screen right here. All right, very good. All right, so let's watch this together, and we'll watch it a couple times because this has got to be like one of my favorite videos in biology, period. And let's see that divide. So this is not starting from fertilization. This is just an interesting video of how the embryo forms generally from like the 200 cell stage. And I want this video to serve as a sort of overall ambassador for the study of developmental biology, because I want to just tease out the fact that the process by which this happens, you see this pulsating, this like this synchronous wave of divisions. This is all stuff that I remember hearing about in class, and this massive cell movement here, right? You get from this wave, this pulsating wave, this synchronized wave of development, into this huge synchronized movement of individual cells. We learned about how that happens as well, and this is a topic that I would really love to revisit. To teach to all of you, because even though this process seems very mysterious, if we take a look at the molecular level, we can actually understand why and how these things happen, right? So this wave of division, mesmerizing. Not only the wave of division, but also the coordinated dance, this movement. Absolutely insane. Absolutely insane. Those gene names don't feel too scientific as a German speaker, but they are probably just relatively old. Yeah, so I went over how genes were named, and still are named. Basically, in the classic days of molecular biology, you just do like random UV mutation, or you add a chemical that randomly mutates an organism, and then you check to see what their phenotypes are. So if we go back to those gene names, the tailless gene, this is a gene that's broken when an organism lacks a tail. I don't know what organism this was discovered in. Giant, this mutant produces a giant embryo. NERPs, I have no idea. Krupel, I have no idea. But each of these gene names has a reason they're named that way. It's just because they mutated that gene. They didn't know what that gene did. They took a look at the embryo and they went, oh, that embryo is kind of huge, so we're going to call that gene giant. Wait until you hear about Sonic Hedgehog. Yeah. I love the fact that it's called Sonic Hedgehog, although it's now officially abbreviated to SHH when you're talking to patients, because no expecting mother wants to hear that their child has a mutant in the Sonic Hedgehog gene. Yeah. Okay. Fly embryo. Yes, exactly. This is the fly embryo. Krupel and NERPs could also be phenotype descriptions. I actually don't know what that means, though. Krupel is actually a word in German. I wonder what it is. Krupel means something like cripple. Oh, yeah, that's probably it then. Okay, very, very cool. No fun allowed. That's true. NERPs means something like someone small. Oh, okay. So we actually do have good reasons for it to be those as well. So I'm guessing that mutating NERPs resulted in an abnormally small egg. Krupel resulted in an egg that looked really, really weird. And then the rest you can kind of make out using the English words. So yeah, that's cool. That's I learned something today. Let's watch this video one more time just because I want to. I think the synchronicity is really, really interesting. And then I promise we'll move on. I just love this video. So cool. Yeah, and again, the reason why this is like final boss level is I don't remember how this happens. I just learned that I learned it at some point and I need to review. Lovely, lovely, lovely video. Okay. So, we've been talking about worms and we've been talking about flies, but what the heck about mammals, right? So the developmental model organism that we have for mammals in terms of developmental biology is the mouse. Mice are incredibly useful model species. They are very straightforward to genetically manipulate, and we've gained a lot of insight into how the mammalian embryo develops by studying how mice embryos develop. So symmetry breaking in mice is not fully understood. At the very latest, the polarity is established at implantation. And so if we walk through sort of the events that happen during the development of the human embryo, you know that sperm fertilizes egg, the egg moves up the fallopian tube and eventually implants into the uterus. And that's kind of a way that you can get that polarity, right? So way before in chat, someone had said implantation as being a potential symmetry breaking cue, and certainly implantation into the uterine lining can provide an asymmetry, right? But what we actually know is that for the mouse, at least, polarity is established somewhere in this region here. That there is already polarity established here. You can already see that there are cells that are clumping on one end of the egg, but actually, mysteriously enough, polarity is already established at this point, somewhere here, before we get to implantation of the embryo. Yeah, so unlike worms or flies, developing mammalian embryos implant into the mother's uterine endometrium, and the zygote itself is not polarized. So while you do get a sperm entry point providing a symmetry breaking cue, even after that sperm enters, that zygote still has you could call it infinite developmental potential, right? It's still very, very potent in terms of the cell types it could give rise to. And so the actual symmetry breaking cue, the part that determines which one of these more or less identical cells become different cells, is established at this stage. Have we gotten to the weird twisting thing yet? I'm not entirely sure what you're referring to here. Does fertilization really happen that high up in the fallopian tube? If it implants too high up, that can be catastrophic, right? Yeah, so improper ectopic implantation can be quite bad, although I'm not well read enough to give you more information than that. Yeah. Things that happen in tissue is often where my weak point is, as longtime viewers of my channel can tell. Like cells, I'm pretty happy with. When we get to tissue, I'm like, okay. So I want to pull from this paper, actomyosin polarization in 2020 through PLC-PKC triggers symmetry breaking of the mouse embryo. So this is a paper in 2017. So this is actually pretty incredible to me because the details by which how the mammalian embryo establishes polarity was being worked out around when I was in STEM graduate school, right? So I think there's a sort of bias when you are young to believe that, oh, you know, we know so much about everything because you're learning things for the first time. But actually looking back, I'm like, oh my gosh, we like figured this out not that long ago, which is really, really surprising to me. It just shows you that you can always continue digging with regards to making up the detail, because cell polarity, which seems like such a basic thing for us to understand, in the mammalian embryo, yeah, it was relatively recent when we got that information, which I think is pretty crazy. 2017, that's pretty old. Don't say that. No. Don't say that. I'm apoptosing live. Oh gosh, 2017 being old, I'm crying. Okay. All right. Okay. This paper actually finds that cell polarity of the mammalian embryo is established at the 8-cell stage. So up until this point, these 8 cells in the developing embryo are more or less the same. And so this is the last time I'm going to ask chat to ponder how you could break symmetry in this particular system where you have 8 cells. For research, that's pretty old. It's old in the sense that, yes, it is old research. However, for such a basic question, it is not that old. For a basic question like cell polarity, right? Like, from a level of like, you know, in 2017, we had already like ran the particle accelerator that allowed us to detect the Higgs boson or something like that, right? Like that gives you a sort of idea of the relative advancement. So it's not that old in terms of our relative basic understanding of how embryos develop. 2017 was not that long ago to answer this very, very basic question. So apparently, if I remember correctly, at a certain point in embryonic development, a group of the cells move in a clockwise fashion 180 degrees, resulting in changes in body lead development. Yes, so there is a point in which you get that rotation. I'm not prepared to talk about it on this stream, but I definitely will when we develop individual streams to these different animals, right? So when I get to development 101, I will have a much better 101 overview other than just focusing on cell polarity. And then from DevBio 101, I'm going to have like C. elegans 101, Drosophila 101, Xenopus 101. So the rotation of those cells does eventually happen, and you do get this coordinated cellular movement to form the different layers of the embryo, the, you know, mesoderm, blastoderm, ectoderm, no, I'm getting it confused, ectoderm, endoderm, mesoderm, etc., etc. Like an invagination of the cells, but we're actually not going to get to that point. We are just looking for the first cue that breaks the symmetry of an 8-cell system. All right, so it looks like a crystal system, tetrahedral. It's my rough drawing. Don't take it too literally here. It's just my drawing that I get to make because I don't want to violate copyright law. A particular point lattice group. Yeah, it does look like a crystal here. When one of them differentiates, the design around that one. So that's a very good hypothesis, Freak, about, and that kind of goes towards like Alan Turing's morphogen concentration gradient idea that like, if by chance one of these pops off before the other, the rest of them can respond to one of them popping off. So that is a very legitimate strategy. That's not how the mouse does it, though. So I'm not saying you're wrong. I'm saying that in this particular case, that's not the strategy that the mouse uses to develop polarity at this particular stage. By how many neighbors the cells have, that is a very, very good answer. It's not entirely right. It's not entirely wrong either. But that is barking up the correct tree. So we won't get to talk about which opening comes first in the deuterosome. No. I'm not ready to talk about that. Not yet. But we will. Not yet. Today we're only focusing on cell polarity. I remember you said to put a pin in cell junctions. Yes, yes. Jtaro, and please tell me if I'm saying your name correctly. Jtaro. Uh, uh, those interactions at the cell junction, that is something that I want you to recall for this organism. So what polarity could be established here, taking into account that A kind of has the right idea by how many neighbors, like that's barking up the right tree. Jtaro has said, talking about cell junctions, that's another piece of this puzzle. This you could actually work out, chat, and I want to see if we can push ourselves to come up with the right answer by taking elements of both what A said and what Jtaro said. Some sides of the cell have many neighbors, some sides do not have many neighbors. Getting closer, but let's just assume that they all have the equivalent number of neighbors. That might not actually be true, but I don't want to get too far bogged down into that particular hypothesis. Let's assume that each neighbor has exactly the same number of neighbors as everything else. If the embryo is moving, the direction of its movement and how nutrients flow around it, also a very legitimate, very good answer. Not exact. I mean, we could discover that that actually matters at some point, but not what this paper was focusing on. Maybe having two proteins that inhibit each other's production and thereby having like a random chance a side that produces more of one. Ooh, okay. A, how many neighbors does the cell have? I'll just say neighbors, okay? Because I want to just focus on the idea of neighbors. Let's take A's idea of neighbors, and then let's combine that with Jtaro's cell junctions idea. Let's combine that with Silas's idea of protein inhibition. See if we can think of something there. Basal/apical. Okay, so connected to the mother versus not connected to mother. So Hairy Day, this is before the embryo actually attaches to the mother, so not quite there. But we have three ideas here that can be combined to form the right answer. Basal and apical, that's jargony. That is the correct answer here, but I want to get there. So if you're a biologist and you know what basal and apical means, then you already have the answer. And if you aren't a biologist and don't know what basal and apical means, I want to see if we can fill in the blank. Yeah, so a concentration gradient will come into play, but let me rephrase the question. Keeping those three answers in mind, what axis of polarity can we think of? Let's name every axis you can think of in chat, and then that I think will help us get to the right answer, because this is super cool and it's not an answer that I could come up with. So I want to see if we can work together to try to figure it out. So base versus apex, that's definitely one. So can you elaborate a little more about what you mean by base versus apex? And then also, like, there's left, right? There's up, down. What else do we have? Left, right, up, down. What do we think? Ah, we have the answer here. Someone in chat's got it. Someone in chat got it. A says outside and inside. Yes. Yes, that is the correct answer here. So when we talk about basal versus apical, what we are actually talking about in this particular case is outside versus inside. Okay, so how does that work? Right, so up and down we can't really do anything with because we're assuming this is symmetrical. Left and right, we can't really split it evenly that way either. Like, there's no mechanism. But if we combine thinking about outside versus inside with the idea of neighbors, with the idea of cell-cell attachments, we can actually see that this 8-cell stage has the potential already to form polarity. So how does that happen? It happens by localizing proteins to the part that is not facing neighbors. So do you see what I mean by combining all of your ideas? We've gotten to the right answer. We can actually figure out, or not figure out, but we can hypothesize that at this stage that there is an outside-inside gradient that is about to be established because we have cells that have neighbors, right? So this cell has contact points with these 4 cells. So maybe that gives this cell a signal to make a protein that is away from that attachment point. So outside facing and inside facing is an axis that we already have the cue to break here. Isn't that cool? So cells know where they are facing the outside and the inside, but I don't know how to differentiate from a perfect sphere. Yeah, from a perfect sphere, it's very hard to determine an axis, but because we have 8 cells already, these cells already have enough information to determine outside and inside. I think that's so cool. Now that that's over, the first A is pronounced the same way as the second one. Jtero? Or, sorry. Can I call you Kemery? I'm bad at enunciation. But yeah, I wanna rest on this idea because I'm very proud of chat for having come up with the correct pieces to solve this puzzle. It's impossible to do up, down, left, right if you are 8 identical spheres with 8 identical neighbors, but you can do out and in because you have one side that touches other cells and one side that doesn't. Kemuri? I can call you Kemuri, I think. Okay. Yeah, so let me know if that's chill. Okay. Yeah, so I think that this is a very clever system, outside and inside. So imagine this. You're a cell, you have neighbors, you're a perfectly symmetrical cell, you don't know what up, down, left, and right is, but you do know that something is touching your butt. So you avoid making certain proteins at the butt contact point, and then you do make proteins on the opposite end. And then you can imagine when you go from 8 cells to 16 cells, when you do a division, one of those cells is going to inherit this blue protein, and one of the cells is not going to inherit the blue protein. So the cells that inherit the blue protein are going to become the placenta, and the ones that don't inherit the blue protein are going to remain stem cells that form the fetus. The beginning of the meat tube form, yes, exactly. All right. So let's take a look at the actual image here. So unfortunately, we don't have a video, but we can take a look at the actual fluorescence of these proteins and also look at that par D6, par proteins, right? So studying the C. elegans worm is useful in our case, right? Because we can actually already guess as to what proteins are involved in this determining of partition. So let me explain how to read this. In purple, we have our partition protein par D6, and in green, we have F-actin. So actin is a component of the cytoskeleton. And we can kind of see here that in the early 8-cell stage, we do not have polarity here, that we have F-actin kind of uniformly around the cell. So what we're seeing is a slice of the cell here, a confocal slice. So don't think of it as like looking directly at the embryo, but taking a slice through the embryo. So this uniform green around the cell and this slice indicates that there is no real polarity going on here. And even though they have different shapes and everything, these are each individually like pretty plurally potent, that they maintain a very high level of flexibility with regards to what they differentiate into. So don't look into that too much. Over here, we have a line that is drawn around the perimeter of the cell to get an idea for how much of that protein is there. So if we go along this distance, if we trace along that perimeter, that is what this axis is, the distance along the perimeter in this direction. Or, sorry, in this direction, in the opposite direction. So in blue here, we have protein levels of F-actin and par D6 in the zone that the cell is not touching. And we can see that it's low, and we can see that at the early embryo stage, we have another segment that is touching a cell, it's also low. So this is the early 8-cell stage here. How do people get both left and middle picture? That's a good question. So this is, I believe, epifluorescence. So this is capturing the light at all planes. And then this is a confocal microscope image. So this is when you are redirecting lenses only to collect light from one particular slice of the sample. I'm not like a microscope guy, so I don't completely, I don't have more than that as my information, but one is epi and then one is confocal. But you could theoretically determine random left-right through small random deviations in signal molecule secretion and then positive feedback. Exactly, that's what Alan Turing is getting at in terms of the morphogen model of differentiation. And that does come into play. But in terms of the first cue, the very first time an embryo goes from pluripotent to having different cell fates in the mouse embryo, as far as I can tell, there may have been more research out there, but from what I could find, this is at least one of the very first cues that an embryo gets to differentiate between different cell types. Okay. Over time, this is the late cell stage, late 8 cells. So we're still 8 cells, right? We have compaction going on, which is another process by which the embryo develops, right? So you go from cells that are blebbing to cells that are very heavily attached, that have very solid cell-cell contacts. And we can actually see in the epi this time that we have a node that the PAR D6 protein has accumulated to. Now, the details by which this happened are in the paper, and I didn't really have time to get too deep into sort of what biochemistry is happening, but we've already seen in the C. elegans a potential way for that to happen is just by this co-inhibition, this mutual inhibition of signaling. So there's some sort of signal that this cell is getting from its neighbors that says, hey, don't put PAR D6 here, and so PAR D6 accumulates on the other end here. And so now if we trace across the cell, we trace across the part that is contact free, and then we head down to the part that is contact full, I guess you could say, we see that there is significant enrichment of F-actin and PAR D6 along the edge of the cell that is facing outside. And the part that is facing inside, the part that is touching the other cell, we get very low levels of those equivalent proteins. And these are insane, right? I think this image is so cool. So again, just to emphasize how cool I think this is, if you were to ask me how I would break the symmetry of this embryo, I'm not smart enough to have come up with outside versus inside, but some of you are, and that's very encouraging. So breaking that outside-inside symmetry by taking advantage of the fact that cells that are in contact with one another can send unique signals to inhibit the presence of certain proteins. And you can already see that now when this cell divides, if it inherits the PAR D, it's going to become placenta. And if it doesn't inherit the PAR D, then it's not. Yeah, so this part of the cell, when it divides into placenta, this part will divide into the fetus. Very cool. Mammalian embryonic development, as you can tell, is very complicated. And again, dreamless sleep is probably more equipped to handle most of the questions as to how this works at an overall level. My focus on this stream was just how does the cell understand its head from its butt? The very first instance that it goes from infinite potential to finite potential. And at the 8 cell stage, that is when you ask some of those very potent stem cells to become placenta, and then the rest of it becomes a fetus. And of course, if you are bothered by the fact that I have not explained how this happens, don't worry. That will happen in an eventual stream where I talk about the mouse in much more detail when I have the time to actually figure that out. Okay. The thing is that the model is simple at first glance, but biology is messy. And this is kind of getting into what a lot of you are saying about the fact that there can be very slight asymmetries that can tumble out of control. And you're absolutely right. That does come into play with regards to cell differentiation and cell fate. So I want to address that a little bit by taking a look at the fact that division, along different planes, different axes are not exactly perfect. So it is very tempting to believe that all of these cells divide in exactly the same way. Like, if this cell divides along this axis, then that makes it very clear that this cell is going to become placenta and then this cell is going to become fetus. The thing is, though, is that biology is messy. And so the splits don't necessarily happen symmetrically all the time, right? So in this perfect asymmetric division, in this case that I'm talking about where the embryo splits, or rather the cell splits here, and then you get the top cell forming the outer cell, and then you get a very clear inner cell, that doesn't always actually happen, right? So you could get a sense that what if this cell split perfectly in half here, and then that division gets you two identical cells. Both of these would become placenta. That's bad. If all 8 of these cells split perfectly in half like this, that would be pretty terrible, right? Because then you would have no cells that look like stem cells. That means no fetus forming. And then you have this case where you have oblique or imperfect asymmetric division, where you get a plane of division that is not neat at all. You get a cell that kind of has an element of both. And then what happens, right? This is all very possible and bit of a headache. And so I want to incorporate what chat has been saying about probabilistic Brownian motion, asymmetric division of things into this particular model. And it's part of what makes developmental biology so complicated, but also pretty intuitive, right? I think biology is very accessible science because of reasons like this, because you can just visualize things, you can just imagine things in your head, and it's not quite as abstract, I think, as math can be, because we could just draw pictures of these cells, right? So if you have a case where you get a perfect halfway split right across the top and bottom axis that gives rise to 2 cells that look like they're going to become placenta, then okay, they become placenta. That's just what happens at in a non-zero frequency. And then if you have a case where you split perfectly down this axis, right in half across the horizontal plane, you get a cell that is completely stem-like, apolar and inside, apolar meaning like completely round, didn't inherit the par D6, that becomes the fetal cells, right? So that makes sense. If you get the entirety of the par D6 membrane part, you become placenta. If you get none of it, you become the fetus. But then there are the cases where things are not quite as neat. Now, there is a hypothetical case where you inherit the outside par D6 protein, but you get shoved into the inside of the cell mass. This actually never happens, so we don't actually have to consider this. So we can just kind of ignore that. But what is kind of a headache here, and this is what this sort of balancing act is about, is that you could get a split in which you inhibit, you don't get the par D6 protein, but you get shoved to the outside. So you look like a stem cell that should be a fetus, but you are in the wrong location to become the fetus. And so, you know, this idea of Brownian motion, this idea of probability, this idea of local probability and determining cell fate, there's a balancing act here where you have 2 forces, polarity, which is sort of the difference in shape, as well as contractility. And so these 2 different ideas, these 2 different states can influence whether or not you get a repolarization. So whether or not you force these things that are in the wrong location, but the wrong location and don't have the correct proteins. If that's the case, then you get like pulled back into the inner mass where you can become a fetal cell. But if you are closer to becoming an outside fetal cell, then you can actually get repolarized. So if you get shoved to the outside, but you inherit enough of it, probabilistically, there's enough of that polarization where you can have further polarization to make it look like you belong on the outside. That's confusing, and I wish I had rehearsed a little bit of how I would explain that, but let me put it this way. The splits here, we're not always going to get a perfect split. Your contribution, chat, into talking about Brownian motion and probability does come into play here, where because there is imperfections in how embryos split, the internal state of the cell will determine which way it gets shoved. So if you are outside, but you should really be a stem cell, you can actually be internalized, pulled back into the inner mass. Or if you've inherited enough of the correct stuff, then you can be repolarized and actually be forced to look more like a placental cell. Does that make sense? Why does polar inside never happen? I don't know. I think in the paper they just say that it's never been observed. So maybe it does happen at some rate, but it's maybe rare enough where it doesn't happen, or maybe there's some level of lethality involved. I'm actually not sure why you never get a polar inside, but it is very interesting. This sounds like it can go very wrong. Yeah, so I'm not like maternal fetal health expert. So I would love to incorporate bits of that in my next charity stream to talk about health with regards to developmental biology, because this is very like heady and model-y and molecular in nature, or cellular in nature. Yeah. So that kind of brings me to the end of the cell polarity Valentine's Day fertilization stream, and I'll just end by summarizing, in case you've joined late, the highlights from what I've talked about today. So cell fates are regulated by the establishment of cell polarity. If you are a perfectly potent stem cell, you have infinite potential, you can be anything you set your mind to. Eventually you're going to have to make a choice. You're going to have to make a choice to become a head or a butt or a chest or a back. And that has to do with cell-cell signaling. And so simple events like the entry of the sperm into the egg can act as a symmetry breaking event. So for C. elegans, the polarity is established by the very act of the sperm entering the egg. And so the way that this happens is that the entry of that sperm causes the membrane to ruffle in a way that it causes this flow of cytoplasm that pushes a particular protein in the egg to the end that the sperm entered from. And that pushing of those cytoplasmic proteins attaches to the membrane that the sperm entered from. And then there is mutual inhibition of the PAR21 proteins and the PAR63 and PKC proteins. This mutual inhibition, this mutual antagonism, they're like fighting each other for space on the membrane, is what sets up a natural breakpoint by which we can have asymmetric division. So for C. elegans, symmetry is broken right at the moment of fertilization. We already know what destinies these cells are going to have. In Drosophila, this symmetry breaking happens even earlier. The Drosophila doesn't even bother with the sperm with regards to the first symmetry breaking event. The oocyte is already pre-patterned with maternal RNA. So as the egg is being formed, the cells from the mother actually deposit mRNA molecules in a certain concentration gradient to already set up the egg for success with regards to understanding its head from its butt. In mice, the polarity is established quite late at the 8-cell stage. So before 8 cells, if you took a look at each individual cell, you're not entirely sure which cells are going to become what in the body, but at the 8-cell stage, you can already start to determine, okay, this cell is definitely going to become a cell of the placenta. And so the symmetry breaking moment here is the fact that there is outside-inside asymmetry already established at 8-cell stage because it has neighbors, right? The side of the cell that touches its neighbors gets one type of signal. The side of the cell that doesn't touch cells gets a different type of signal. And that already sets up an inside-out asymmetry that allows for asymmetric division. So the cells that divide and get a lot of the purple stuff are going to become the placenta, and the cells that don't inherit the purple stuff are going to stay on the inside and become the fetus. So despite embryogenesis being literally the most important process in an organism's life, it is literally the beginning of the formation of an organism. The mechanisms are surprisingly diverse, right? So the reason why this surprises me so much is because this is so central, right? Like this has to go right for an organism to even play the game of evolution. And the fact that we've covered 3 different organisms with very, very different ways of establishing polarity is fascinating to me, right? There's a very central idea of just how does a cell know its head from its butt is very, very different depending on if you're a worm, a fly, or a mouse. And there's a part of me that thinks that it shouldn't be. The fact that like you need this to just play the game of life, that it should be more conserved, it should look the same across different species, and it's not. And that brings me a lot of joy because, I don't know, when things are unexpected, there's always a bit of intrigue or happiness that I get. Being surprised is always such a joy in biology. All right, that's the end. I'll just end on this slide, I guess, and hang out for a bit while we talk about whatever for a bit of time. So, chat, what do y'all think? I know. Maybe I oversold the stream. Like, I definitely feel like there were people who were expecting me to talk a little more about the development of the embryo, and I will. It's just that it's a lot. It's a lot of work. Embryos are complicated, and this was also like just to talk about stem cell school for a bit. Just a brutal time in stem cell school is studying for the exams here, because you pretty much have to recapitulate how you go from egg to organism in not only just these three. I picked the three that I remember the most because these are my favorite, but we did zebrafish, we did frogs, as I had mentioned earlier. What else did we do? I think there's one more organism that we had to learn whose name I'm utterly forgetting. But yeah, and each of those organisms has like a different lesson as to how cell polarity can be broken. And what's relevant there is the fact that by understanding how cells can break polarity, we can check to see if they do it similarly in other downstream applications. So cell polarity is not only a thing in developing embryos, but also just your cells in general. And cell polarity also comes into play a lot in cancer. This has broad functionality in biology, and I really want to get into how that is established, just because there's something very pleasing about looking at an embryo or looking at a cell and being like, there's a head and there's a tail to that cell. It's very, very neat. Oh, did I not set up the Discord link? That is super unfortunate. I had meant to. Let me go ahead and just paste that in the chat. I had meant to set that up with my stream elements thing, but let me go ahead and plug that. Oh, gosh, there are so many other things to do for the stream. Here it is. Sorry about that. Here it is. Embryology is one big burger, yeah. Awesome summary I got here at the end of the mouse explanation, but I got the gist. I think at least that's good. That's good. It was never going to be like all of mouse embryonic development. It was always going to be very specifically just cell polarity, just figuring out how head work from tail. Okay. I enjoyed the stream. Wish I caught the start. Oh, no worries. There's always the VOD. Yeah, Homo sapiens is an interesting organism to make a video about, just because I think that I will personally stay in the realm of mice and then just trust that things happen the same way in humans, just because I don't want to talk about human specific stuff, because as you can imagine, it is a bit of a moral hotspot for certain people. Yeah, it's like a semester, exactly. Like there's no way that, just streaming for fun I could cram all of the information about embryology into one stream. Like, I just don't have the RAM in my brain to have figured it all out. But I think that's actually a benefit, in the way that I also want to show that I am learning along with you, that because professors often have this like very high and mighty kind of vibe, which they've earned because it's literally their life work. But me, I am there in the trenches, and I'm excited to bring you what I've learned from the trenches of Google Scholar. Put it that way. How are stem cells characterized? You can't just stick a pluripotent stem cell anywhere and expect it to just work, right? So that's a really interesting question, because in stem cell school, I learned that you kind of can, depending on how pluripotent your system actually is, you actually can just add, like, you can do experiments where you take a pluripotent stem cell and put it in a different embryo and see and trace what that cell becomes down the line. There's a lot of stuff that happened like that in the study of embryology that allow you to do that. And that's the kind of stuff that I really need like a ton of time to dig through papers to find those experiments, because I think they're super cool. Because you can like graft pluripotent stem cells onto developing embryos and just see like, oh, if I get another thorax or whatever, then that particular pluripotent stem cell is what becomes the thorax. Or like, you know, if you take a pluripotent stem cell and it only becomes thorax 100% of the time, you know that that cell fate has already been determined. I'm butchering the actual experiments, by the way, but that's kind of the gist of like what I've learned. There's a set of experiments that you can test the stemness by removing it from its regular environment and seeing how stubborn it is to change. So yeah. How does the observed phenomenon of the teratoma fit into this? Oh, that's a good question. I should do a teratoma stream one day. I think that would be cool. Just to give myself an excuse to study teratomas. From what I understand, it's more about a reversion to a more stem-like state. But I don't actually know, because so for those of you who don't know in chat, a teratoma is a type of, I want to get this right, a type of tumor, I think, that can differentiate into things like teeth, hair, which is really creepy. And I don't actually know how teratomas decide, quote unquote, how to become teeth or how to become hair. So you could just have like a thing growing in your reproductive tract, for example, that is growing teeth, and that is scary, but from what I understand, you can actually deal with it. So it's like freaky from a body horror point of view, but that's why we do science, right? To help people with that kind of thing. So like chiral organisms. I'm not entirely sure what that's a reference to chirality in this particular application. I was more thinking about during the adult phase. So like adult stem cells, right? So I am only familiar with adult stem cells in the niche of the like brain, the fact that there is a limited population of neuronal stem cells that become neurons. Yeah, so I'm not going to be able to speak too much about that. But what I do know is that there has been really good work in the lab on how to grow neurons that are more neuron-like by forcing cells to become more stem cell-like, so that they actually become more neuron-like instead of getting like a tumor. Because a lot of historic studies in neurobiology have been done with neurons that are tumor-like, like cancer cells that have been forced to look like neurons but don't actually share that much in common with neurons, which has been a sort of flaw in the neurobiology literature. So ideally, having a real neuron stem cell become a real neuron would be really cool. But that can be kind of hard. But there's been good work to try to make that happen. Can we just get a teratoma podcast? I don't need the pictures. That's true. If we ever have a teratoma stream, I will for sure have a warning on slides that have the teratoma on them. I've actually never looked at a teratoma myself because I'm, despite being a biologist, kind of squeamish with regards to body horror stuff. Which is also why it's good that I never became a medical doctor. I'll just put it that way. Stemness seems a bit vague of a descriptor. There's more to it, just more or less of it, isn't there? So stemness is a way that the concept gets simplified, and for my background, it's been good enough. So I'm certain that it's more complicated than stemness, but for the purposes of having a general cell molecular biology education, I think stemness is an okay way to simplify it. You're a neutrophil. What about hemopoietic stem cells? I'm a neutrophil who's still learning a lot about myself. We'll just put it that way. You know, across the multiple lives that I've lived, there was a situation where I would have differentiated into a neuron, although you could argue that's the wrong niche. But yeah, my background is in neurobiology. How did you get into biology first? I wanted to be a medical doctor because my parents told me that I should be a medical doctor. And then eventually I said no. I was a very good, well-behaved kid. I didn't rebel against my parents at all. So they were like, you need to become a medical doctor because you make money that way. And I said, okay, you got it. So I very earnestly lived my entire life thinking I was going to be a medical doctor without like a shred of self-doubt. And then I got into undergraduate, and then I had my first physiology class where I got a B. And the thing about getting a B in physiology is that you need a very high GPA to get into medical school. And getting a B in physiology was the first sign that maybe I would not be a good doctor. My memory actually isn't very good, you know? It's often said in high school that like you need a really good memory to be a biologist, and you do to get a high grade, but when you get to upper level biology, you need a good enough memory. You don't need a great memory. But for medical school, you need a great memory, right? 200 bones, 1,000 muscles, you know, all of these things that you just have to memorize to make it through medical school. Getting a B in physio was a real wake-up call to be like, I do not have the gigabytes in my brain to have that much information in it and be able to recall it. And you kind of want your doctor to have a very good working understanding of how all of that works, and I just don't. That is to say, like, I had a talk, and I've told this story multiple times, so it's worth talking every time just because there are more people watching. I had a conversation with my pre-health advisor, and he was like, what do you want to do? And I was like, I want to be a doctor. And he went, why do you want to be a doctor? And I went, because I like biology and I like helping people. And then he said, why don't you do research? And then I said, that's a great idea. He said, do you like working with patients? And I said, I've never had the chance to work with patients before because I've gotten rejected from every intern application I've ever had. And he said, well, your life is going to be mainly working with patients and not a lot of science. And I said, oh, that's no good. Dealing with people who are sick and angry all the time doesn't sound very pleasant. I don't think I have the demeanor, the sociality, the discipline to be a good medical healthcare professional, because it's multifaceted. Having known doctors in my personal life, like, it's not just you have to be smart and you have to have things memorized, you also have to be able to work very well as a worker. Like the things that make you a good worker also make you a good doctor. So the ability to collaborate with your peers, clear and efficient communication with your hospital, with your job, with your patients, it's a lot of that. And of course, bedside manner, all of that becomes like a very multifaceted, deep job. And really, I just want to read science and yap about science. That's kind of my thing. I would love to yap about the science to a patient who is sick, but something tells me that they want to learn how to get better and not how cool the underlying biology is. Having a passion for biology makes learning biology easier, but it does not make for a good doctor. It's not, I should say, it's not sufficient to become a good doctor. I'll put it that way. Hope for me yet for bio. Yeah. I mean, my memory, like if you tell me a phone number, I'm not going to be able to memorize it. You know, there's a concept called like working memory, where it's a vague concept of how many items you can store in your head at any given point. And I've got like a working memory of 5. You can actually test it online by like getting a series of numbers that are flashed before your eyes. People range between 5 and 7. I'm on like the low end of 5. I actually can't cram, and that's what made grad school so hard, I think. Like, I used to talk to my classmates and they were like, yeah, you know, the paper was so hard, I spent like 4 hours on it, it was really confusing. And then in the back of my mind, I was like, 4 hours? Dude, I spent 8 hours on that same paper. And in graduate school, I realized that the reason why I spent 8 hours on that paper is because by the time I finished sentences, I would have already forgotten the first half. Which is devastating, right? Like, that's how kind of short my working memory is. But the thing that gives me hope, or the things that I think I want to explain that should give you hope, is that learning itself is a skill. It's a metacognitive skill, and it's a skill that gets honed the more you do it. So because in graduate school we were reading like 4 or 5 papers a week, that just forced me to get better. You do get better over time. So even though my working memory is very bad for things like numbers, for biology papers, it's like riding a bike, right? There's a fluency that accumulates, and I no longer have that issue where I've forgotten the first half of the sentence before I reach the last half. What else? What else? What else? Skipping out on patient interaction is why I chose the job. I chose within the field of healthcare too. I can relate. Yeah, yeah. Like, I know that I think what makes for a good doctor, and far be it from me to tell people what makes for a good doctor, but if you're like me and you like biology and you have an idea that you want to work with patients, do it for a little bit. Work at a hospital, like apply for every hospital you can, and just be in a patient environment. And if that sparks something in you, if you're like, you know, the patient's face when they light up and they tell me they get better, or I want to be the one to break the bad news to a patient because I think that I don't trust other people to do it because I know that I can do it very well. You know, if you have that kind of spark where you have patient interaction as a central core theme to what you find enjoyable, then you have my approval, right? Whereas if you're just like me, you like biology and you like talking about biology, that's really not a good enough reason to be a doctor. I want to be a medical doctor, but my undergrad is bioengineering. But that's perfectly fine. Bioengineering, actually, there has been a recent push for more engineers to become doctors. In terms of thinking about diversity in the workplace, it's very useful to have a diversity of training, right? Like an engineer thinks of problems in a very different way than a biologist would or a scientist would, right? There is a different skill set. And there has been a recent push, at least in America, to hire more people with an engineering background for doctors. So that's not what I will say is that engineers have lower GPAs, which I think is a misconception that it hurts that much. You have to have a good grade no matter what. But if people see that you're coming in from engineering, from my understanding, they're likely to be a little more sympathetic because engineering is a harder major than science is. Generally, it's not universal, generally. Off topic, but when probiotics stream, right now probiotics are a bit annoying to talk about for various reasons. So I already did one with myelin, actually, so you can check my archives in the live tab of my channel if you want to hear more about probiotics. But right now, not a good time, I think. Yeah, me too. I have no RAM. Yeah, yeah. The RAM is the best way to think about it. It's just overwhelming, the amount of information. And don't get me wrong, in graduate school, biology is also overwhelming, right? I remember feeling so like, damn, my peers can read a sentence one time and understand it. But what carried me through is my absolute love for the subject. Like, it was just brute force, gritting my teeth and sitting down and burning through uh it's dramatic to say, but like years, maybe months of my life, in terms of like day, like grad school was, those classes were hard and stressful. But thankfully I only had to take 1 year of classes for my program. Yeah. Yeah, I really think I should do stem cell stuff. There was a big emphasis on stem cell biology back in my undergraduate, but not that much in my program where I ended up. So it would be a good way for me to revisit that literature, the literature of stem cell niches and stuff like that. I think that'd be a lot of fun. And probably answer a lot of people's questions about stem cell research as well, because at least when I was growing up, that was a big issue. Um, and also like, yeah, just, yeah, and it also dovetails very nicely with what we've already talked about with cancer, this idea of stem cell-ness. So if you've watched my 12-hour charity stream, there's this idea that reversion to a more stem-like phenotype is characteristic of a lot of cancers. And so for me, if I were to like tier list my own biology interests, I'm more interested in stem cell biology for the application to cancer than I am for something like regeneration. Um, which would be interesting to get other scientists on, because like there are people who are obsessed with this idea of regeneration, and like I'm just not totally sold on it personally. And it would be fun to have another scientist on who is doing regeneration to do that kind of stuff. I think there's very, very good potential for that kind of thing. What's my favorite cell in the human body? It's a cop out answer, but it's the neuron. I might be a neutrophil, but I have a deep appreciation for neurons as cell types, just because they are what I studied in stem cell school. So neuron, easy favorite. Because they're like little nodes, right? Little circuit machines that convert electrical activity, and they have an output as well. So there's a very like STEM-y nature, not STEM as in like stem cell, but STEM as in science, technology, engineering, and math aspect of them. And they can communicate with one another. They have interesting uses for immune proteins, although you might argue that that's not necessarily a property of neurons themselves. But they're also just really pretty to look at underneath the microscope. Bunch of neurons says neuron, shocker. Yeah, exactly. This is neuron propaganda for sure. But yeah. If you haven't already, I've linked a document in the pinned comment. If you have a couple minutes of your day to nominate me for best lore VTuber, that would be pretty chill. It's an interesting way for us to assess. So beyond just me, right? Like I have, if you type exclamation point award or something, we're all up for these categories in vtheorem. And it would be really cool to see any of us even shortlisted, just to give us an idea of how much, what our relative position or whatever is in the grand scheme of small VTubing. There's not a small chance that we get something, considering a friend of the channel, Phaedra Sarah, had won a category of a different small VTuber award a couple months ago. Phaedra Sarah being an excellent biology yapper. Who's also more into the medical side of things. So, even though we both talk about biology, Faye does so from more of a medical standpoint, whereas I'm doing it more from a basic science standpoint. So it's not like if any science communication VTuber made it on the short list of that award, that would be a really nice thing to celebrate, I think. So I'm up for the best lore category if you wanna donate some of your time there. Learning about how neurons and how synapses work is a blast. Yeah, like again with this infinite onion of biology idea, you keep on peeling back layers, and the more detail you learn, the more the mystery stops being a mystery. But then you peel back the onion and there's just more mystery underneath that. But every time you do peel back that layer, there's a sense of like, I feel like my brain like exhales, right? Like the first time learning about long-term potentiation and long-term depression, these are the chemical processes by which neurons strengthen their signaling and weaken their signaling as the molecular mechanism by which we learn. Because up until you learn about LTP and LTD, you get this vague idea, oh, neurons, they get stronger, some neurons get weaker, and then that's how you learn. And it's like, cool, okay. As a concept, that makes sense, I guess. And then in undergraduate, when you first learn LTP and LTD, you go, oh, underlying all of this abstraction are actual molecules that are doing things. And those molecules are mediating that kind of learning. And to me, the synapse has always been such an interesting and fun things to learn. Recently read a book on artificial life. One of the main topics was emergence in seemingly simple systems and how that also applies to biology. It was fun. Yeah, abiogenesis is something that I think we talked a little bit about in the Discord. Was it in the Discord? I'm trying to remember. Oh no, I was replying to a comment on Reddit actually about abiogenesis. Yeah, I don't think I talked about it in the Discord, but sometimes I look our biology and our evolution to offer sort of my take on biology communication, because talking about abiogenesis and evolution is very hotly contested depending on your spiritual beliefs. And so I want to sort of offer answers to people who are, we'll say, abiogenesis curious that might offer a level of spiritual satisfaction for them as well. Because if somebody wants to believe that a supernatural power spawned life on Earth and that idea brings them comfort, there's no way you can convince them otherwise. They want to believe that. And they have the right to believe that, you know. But in terms of like debating, there's no sense of debating if the idea is attached to a level of sentimentality, right? And I'm not saying that that's wrong, right? Because I can't disprove that we weren't just spawned on planet Earth. But what I can say is that there is an alternative hypothesis that has evidence, but not a complete picture, but evidence enough that you could fill in the blanks with your mind. And that field of abiogenesis, you know, hypothesizing and postulating how complexity could have emerged is very, very genuinely interesting. And I recommend that if you do talk about that kind of thing, that you do couch it in language that is a little spiritual, just to have a common cultural touch point. Because as we've seen repeatedly in the field of science communication, you do not win people over with facts and logic. You win them with providing a better story. So I think in my Reddit comment, I had talked about abiogenesis, and I wanted to make very clear that chemistry was rolling dice for millions of years until it finally rolled exactly the correct ingredients for life to happen, as a way to kind of couch it in this awe and wonder of how life could have formed. I'm not like an abiogenesis expert, but to me, the idea of chemistry rolling the dice over the vast chemical reaction bath, like the biggest chemical reactor on the planet Earth being the ocean, you know, with that many reactions going on for that much time, it's not tremendously hard to believe that at some point in the ocean, chemistry rolled just right for all the right ingredients to happen for a protocell to form. And I think that's very fun. It started with alien contribution. That's also possible. People have postulated extraterrestrial origins of life as well, or at least donation of biological material from space. Although I think that might be falling out of favor, but I'm not entirely sure. The Darwin aspect of like speaking the language of one's audience. Exactly. It's really miserable, I think, to argue on facts and logic when it's just more, it's more fun to just tell stories. B, it is not how people learn outside of the classroom. Like, I think that what makes university more fun of a learning environment than K through 12 in America, can't speak for all countries, but I can speak at my own experience in the school system, is that in university, your lecturers aren't actually meant for you to learn. And that sounds crazy, and I know it sounds crazy. The pedagogical role of lecture is not to teach you. It's to give you context for you to teach yourself, because the ideas are that complicated, right? Like programming, for example, your programming professors, or at least mine did, gives you the start of teaching you programming, and then it's up to you to practice. They're not, you know, you sitting in lecture, and this is why like gifted kids in high school crash and burn in university. Like, this is the actual difficulty spike. People talk about the difficulty spike all the time without identifying what makes university harder. So this is my little spiel on the difference between high school and college for gifted kids. In high school, if you had a good memory and you could make logical connections pretty seamlessly, high school is not that bad for a gifted kid, quote unquote gifted kid, right? But as soon as that gifted kid gets to college, the ideas actually get so complex that you no longer can rely on your professor teaching you everything. Your professor doesn't have the time to walk your hand through the skill that you need for upper level learning. Because in high school, like, learning itself is the skill. Like, you just learn to learn and then you get better at learning. But in college, you need to learn the skills of a biologist, and you need to learn the skills of a programmer. You need to learn the skills of a physicist. It's that level of specialization that makes it so that your professor cannot teach you everything in your 2 hours a week lecture. It's just impossible. That's why you have extra like recitations or precepts, depending on what universities you go to, sessions where you practice those skills. And so gifted kids come in, they think, oh, I'll just learn from lecture, go home and play League of Legends all day and I'll be fine. When in reality, if you just did that, the things that you get tested on are way more in depth than what the professor says out loud in lecture. So you cannot rely on lecture for all of your learning. And so this is both good and bad. It's good in the sense where it's fun to listen to lectures that are more geared towards stories, because that's how you learn the basics of what you're supposed to be teaching yourself. And then it's bad because if you do no work outside of showing up to lecture, you're going to have a bad time, pretty much. So why I think university is more fun is because they're not focused on walking your hand, taking your hand and walking you through everything you need to learn, but rather, here is the field. This is how you get started. It's up to you to do the rest. And to me, and of course, that I'm not making excuses for bad professors, but that is kind of the underlying pedagogical framework by which lecture operates in university. And I feel like that doesn't really get talked about enough. Oh yeah, Miller-Urey. Miller-Urey is a big experiment that kind of gives you the tools to make connections, to give you the idea that abiogenesis is even possible. But people who want like a very solid, I want to know exactly what happened, you're never going to get an answer because we don't have a time machine, right? Like we don't have enough time. We don't have a reactor big enough to simulate millions of years. Although you could argue with AI, maybe you could do it in a computer. But I don't even know if people would accept that, which is why it's not useful to talk about, oh, they'll believe the way that I believe if I just gave them the data. They won't. It's the story that matters. I believe panspermia just pushes the problem further. Like, how did those life forms and molecules form the original planet? Yeah, and you kind of don't want panspermia to be true because there is like a hard block on how much you'll be able to learn about how life formed. So while panspermia is like a fun idea, it's not necessarily the one I want to be true, just because then that story I think is pretty late. It might be cool, but if you really think about it, it's also not that cool. Not that satisfying. You get used to being the smart kid in your class, and college is full of other smart kids, and you start putting yourself down. I went to a high school with a bunch of smart kids, and so I was already down by the time I got to university. It's kind of like my parents did their damnedest to move me to school districts that were like incredible, because they valued the value of education very, very highly, to the point where they would uproot their lives in order to put me in a neighborhood with a good school, which is a privilege, right? Like I had an incredibly privileged upbringing where I never went to a school that was rated underneath anything less than excellent, essentially. And that was great in the sense where my personality as someone who loves learning, it just happened to work very well for me. But it came with the side of the worst imposter syndrome you've ever seen, up until I got to undergraduate, where it started to get a little better. Because like being surrounded by geniuses all the time and then being mid in a class full of geniuses despite you trying your hardest is like very, very demoralizing. It's like I'm busting my ass and I'm seeing that the ceiling is so much higher than myself, which did a number on my psyche, right? But when I got to undergraduate, like I went to a pretty good school for undergraduate as well. But like I went to like little freaking genius schools beforehand. I went to little genius school. Like I went to genius schools without being the genius, right? Like the cream of the crop were so far beyond my ability back in high school. I was still like pretty good, right? I have to acknowledge that like I busted my ass in school my entire life to be pretty good, but going to a school for geniuses, essentially, while not being one of those geniuses, was pretty insane. It was a pretty insane experience. I googled vitamin V to search for the official landing page of the awards. Big mistake. Oh, is there something controversial by googling vitamin V? I have not actually, to tell you the truth, on finding the landing page. But yeah. But I think that it all kind of normalizes when you're an undergraduate. Then, like, what got better from me there is the idea that when you are doing research in a lab, that you are the expert in that field, and that was incredibly validating. That, like, even though I'd been like mogged mogged the entire time by geniuses my entire life, like, even those geniuses don't know what I know about the protein kinesin, right? Like, that made me feel good, because I got to specialize, because I got to differentiate, you could say, away from the infinite well of potential that young students are. And that little bit of differentiation gave me enough of a sense of identity that I could hang my hat on that identity as, like, okay, sure, I'm surrounded by geniuses all the time and they're really, really smart and everything, but they only have a certain number of hours in the day. This is how I overcame imposter syndrome. Like, there are a finite number of geniuses out there. They have a finite number of problems that they can tackle, and there are more problems out there than there are geniuses. So I don't need to be a genius to work on problems. So I get to exist and justify my existence that way. Is that the healthiest way to think of things? Maybe, maybe not, but you know. Oh, it's Viagra. I didn't know that. The one time I make it to stream and it's towards the end, I'm sorry, Gabriella. But you know, the VOD will be up if you want to look at the VOD. Nice joke, +2. But yeah, we're just chilling now. We'll chill for a bit longer and then I will call it a stream. Yeah, we're talking right now about I went off on a tangent about going to a little school full of geniuses, and how tough that was. But in undergraduate, I had the reverse effect of many, like, gifted kids, because a lot of gifted kids come in, they're like, oh, I don't need to study, and then they get wrecked. And they're told, right, like, in culture right now, it's actually very clear that the gifted kid thing happens, and yet there are still gifted kids out there who think that it won't happen to them, and it will. And it's not because they're not smart enough, it's because fundamentally you don't get enough from lecture to succeed, because you can't. There's too much. It's too hard to fit in lecture. Sure, if you had an 8-hour lecture that taught you every single little thing, but your professor doesn't have time for that, so that's just not going to happen. There's a fundamental flaw, and you're going to have to study on your own because you just don't get that information. My imposter syndrome began towards the end of college, together with a bunch of other personal problems too. Yeah, it's definitely, I think I've definitely had a unique aspect of that where my imposter syndrome kind of let up by itself, whereas I think a lot of people, it's a lot harder, I think, for a lot of people due to, like, my unique life circumstances. I've actually talked to other vtheorem members a lot about the idea of imposter syndrome, and it would be fun to have a somewhat casual stream, somewhat academic stream about the study of imposter syndrome. You know, we do have 2 psychologists on board in vtheorem. It'd be very interesting to learn about the psychology of imposter syndrome, and to share all of our imposter syndrome stories. One story that I like to tell about my advisor is that they had, you know, incredibly brilliant advisor, like, you know, teaching at some of the best institutions in the world, was talking about their imposter syndrome story, and it stuck with me, and it really made me not feel alone, where, you know, they were defending their PhD thesis, studying at, like, an Ivy League, like one of the best Ivy Leagues in the country, and they had overheard, you know, they were at the top of a spiral staircase, and at the bottom of the staircase there were people talking about the defense that they had just witnessed, and they were like, oh, you know, it was so good, and, like, damn, the talk was so interesting. And how I know that part of imposter syndrome is rooted in irrationality, or it can be rooted in irrationality, is my advisor had said that even though that they were hearing these 2 people, these 2 strangers who were geeking out about how cool the talk was, there was still a part of them that was thinking, oh, they must be making up, they're just being nice, they're just, you know, they're not even talking to them, right? They're talking in private amongst themselves. They're eavesdropping on a private conversation about how awesome and how cool that research was. And there was still a part of them that was negging them, essentially, to think that they didn't deserve that kind of accolade. And it's a brutal story. I tell it every time imposter syndrome comes up. But I tell it because it's a sign that your imposter syndrome could have an irrational element to it, which is not necessarily helpful for all people to hear that, oh, the imposter syndrome just in your head. That's not really what I'm trying to get at. It's the idea that, like, your current brain state, like, there's a level of intellectual humility to acknowledge that, like, your current brain state might not be accurate. And it's not easy to have that humility. I know I've been victim to doubling down stubbornly on things that are wrong. But, like, that story to me is one that I think of a lot in terms of imposter syndrome, to try to explain that, like, yeah, even like brilliant professors who have graduated from Ivy League institutions have imposter syndrome, and the way that it manifests isn't rational, right? They're objectively very, very successful, but because, like, by looking at them and then looking at yourself, you can see that even if you aren't as far in your career as them, like, oh, this happens to people who should not have it happen to them. Maybe there's some flexibility there. That's why therapy is important. Exactly. What's your favorite science website or tool? Sci-Hub, Wolfram Alpha, so on and so forth. I think there's a website called OneZoom or something like that, where you can get a sense for the God, what is it? Dang. There's a website. I'm getting different websites confused. I think it's OneZo or OneZoom or something, where you can get the entire phylogenetic tree of life down from the most common ancestor. Let me actually see if I can link it in chat, because it's a very, very cool resource that I love very much. I would love to. Tree of Life OneZoom, is it? See if it's yes, it's OneZoom. So let me go ahead and link that. Put it here in the chat. You could spend quite a bit of time here in the OneZoom Tree of Life. And the reason why I really, really love this website, I don't know if it's my favorite, but it's the one that immediately came to mind when you talked about favorite science website. And so this is the phylogenetic tree. You can take a look at where we are in terms of our relationship to every other organism. And, like, zooming out on the tree, you can really get a sense for the fact that we are all quite related. And you can also get a sense for how we evolved, right? Like, branching off of like mice, you have primates, right? So you can get a sense for how closely related we are to our evolutionary cousins. And the fact that it's like a fractal pattern, of course, fractals being a very important part of vtheorem iconography, very important part of science in general. Fractal patterns are everywhere, and they're very cool. And yeah, you can keep zooming out and just see how we are related to every other organism. To me, that is a very beautiful representation of our place in our world, right? And not only that, but also when you inspect sort of this tree of life, you get a sense for truly how many worms there are out there. Like, someone had called it meat tube, the meat tube body plan, and that's so true. That's so true, fam. Like, in a different universe, we'd all be just worms. Like, we were all worms for a gazillion years, and then a subset of worms just ended up being more complicated, and then that gave rise to, like, every animal that isn't a worm. Right? Like differentiation from the worm body plan is so central to I mean, no, we all have the worm body plan, you know, or animals, I mean, right? But in terms of complex life, we branched off. It was worms in one branch, and then it was the rest of us. And that, I think, is wonderful, I think. Yeah, so that's a pretty fun program. Would you love us if we were worms? The eternal question. Of course I would, surprise pants. Of course I would. Worms are fantastic. We are tubes, exactly. Meat tubes. Meat tubes. I should really publish the meat tube rant that I had. So as a part of the microbiome video, I had like 7 minutes of footage that I ended up not using because the video was getting too long, about the evolution of the microbiome of the gut. And part of that was just saying meat tube over and over again, because the hypothesis that I've seen is that the microbiota evolved in our meat tube form, you know, back when our body plan was that simple, and just happening to have bacteria that are localized to that meat tube. And of course, some bacteria offer a selective advantage and then also offer a selective disadvantage to harmful bacteria, etc., etc. And then over time, you get an interdependent relationship between the gut biome and the organism. And that's a fun story to think about. Would the neutrophil eat us if we were worms? His eosinophil friends are on us wormies. Yeah, exactly. It's not my job to deal with wormies. It's the eosinophils. Patrikios did a video on the immune response to multicellular worms. I don't like, my motivation to get videos out is just like, I don't want to be poached by Patrikios, and I'm obviously never going to be 100% on the ball to get to topics before they do. Not that it really matters, because, you know, Patrikios' audience is huge and my audience is growing, but I think there's very little chance of people, audience members that I care about, being like, oh, Patrikios did it first, although they totally did on my rabies video. Too similar to Patrikios' video. I'm like, I don't know, it's the same virus, you know, there's only so much that we can differ. Like, what is too similar between a Patrikios video and what isn't? There was drama back in the educational content creator space. Not drama, like every content creator was very cordial about it, but like there was a series of content creators who were like, yeah, we scoop each other all the time and it's normal because the topics that we cover are just facts and no one has dominion over facts, so chill with the accusations of plagiarism. And I think since then, like, no one's really been that crazy about like, oh, Patrikios did it first. At some point, I was obsessed with embryogenesis since I saw the hypothesis that deuterosomes have their heads backwards. I actually didn't know that. I know very little about the development of organisms outside of what I was taught in graduate school, so it might be interesting to expand my horizons a little bit. I like the details in your videos better. Patrikios doesn't always go into detail on the biological stuff. Yeah. Patrikios, I mean, it's different niches, right? Patrikios is amazing for like an entry point, and they're very well researched. Of course, they have a team. On Team 5 and Neutrophil, it's me on research, editing, scripting, and then Toxo on asset creation and proofreading and checking the video and everything. Like, it's just the two of us, and of course we aren't going to be able to out research a whole team of scientists, but what we can do is attempt to provide parts that they are likely too afraid to include because it might alienate people, and that's okay. Like, if I think about YouTube as being its own ecosystem, Patrikios is like, I don't know, a shark or something, and I'm like a little parasite. I have a happy and good life, leeching on the environment created by the shark, right? Like, I'm happy to take people who maybe watch Patrikios to get a brief overview, or maybe are biology pilled by Patrikios and want to know more, and then I'm happy to be like, you want more? I got more, and then I'll show them the good stuff. So I'm very happy to be in the same space as Patrikios. I just, for my own scientific competition vibe, because I have not had, like, so backstory. The reason why I didn't think I would be a good doctor is because I had no like, I just wasn't interested in patient interaction. I quit graduate school because I don't think I would have made for a good researcher, because part of being a good researcher is having a competitive mindset. Like, you want to be the first, you want to, stay up late and be the first ones to discover something. If you have another group, another lab that is going to publish before you, you want to try to see if you can cut in line, because that's where the glory is, right? And everyone does it. Of course, you want to do it in a way that isn't toxic to your scientific neighbors to burn bridges for collaboration, right? But in terms of being the first to a thing, there's like rules of engagement, right? There's a level of competitive that you can be where you're still friendly. And I didn't really have the competitive drive to be a good researcher, but in terms of YouTube and teaching, I do have that competitive drive, right? Like the drive to be like, I want to be the first to like make a big video about prions. And I think I did a pretty good job. There are other prion videos out there that are very full and complete, but I'm genuinely very, very happy. And it's a sign that you can see that my imposter syndrome has mostly faded, that I'm quite happy in my own membrane, that like I'm genuinely very, very happy to have what I think is the best prion video on YouTube. It's a very small niche of people who care about prions, but for people who are looking for information on prions, who want to know about the juicy, interesting stuff, I think my video is the best. And I'm very proud of it. And so competition with Kyrgyzstan is like, I want to be the one first to the punch, even though it doesn't really matter, but I just want to, because it's fun to exist in this space. Remora to their shark. Yes, mutually beneficial. Exactly. Like, I might be feeding off of them, but like by keeping people interested in this environment, by keeping them curious, it's good for all of us, right? Like there's no reason to display behavior where you're trying to monopolize like a certain niche. Like you can't. There's this interesting thought that like someone brought up to me. It's this idea of like nucleation of storefronts. There's this idea that, oh, if a sandwich shop opens across the street from your sandwich shop, it's going to hurt both of you because there's competition. But also there's this idea that like if there are enough good sandwich shops that open in the same location, then that area becomes known for its good sandwiches. Then people return to that area to try a different sandwich. You actually help each other build your own individual audiences. I don't know if that concept has a name, but I've always found that idea to be quite fascinating because, yeah, like now that I think about it, like, so I'll take boba for an example. If you're in, say, an area of a city and there's one boba shop there, there's no real incentive to go there if what you're looking for is good quality boba, because there's no competition, right? And so competition arises, and that competition, again, in like just like life, right? Just like Darwin would say, like that kind of competition drives the competitors to get better, right? This capitalism 101 kind of deal. Opening competition nearby, to a certain extent, it does split your audience base. But if you are all good enough, if you all benefit from one another good enough, if you all have like a strong enough presence, you actually improve your business by having competition, because that region becomes known. Your niche gets established. You can draw in people who are looking for science, and then they find everybody else through you. And I think that's wonderful. I think that's really nice. Hey, Mylan, good morning, good morning. Mylan was up late last night rigging a VTuber model for Cideray. So if you ever wanted to see the process behind, what goes into making one of these guys, you might want to go check out Mylan's VOD. It was very, very fun to watch, which is very, very cool. Rising tide lifts all boats. Yeah. Ain't diversity the beauty of lady science? Exactly. It's wonderful. And like I talked to Clockwork a lot about this. The fact that like I actually thought that when I was starting this channel that I would have it look a lot more like Clockwork, but I'm actually a I'm actually a dog water animator, as you can tell by my like very simple animations that I do on this channel. So I'm very happy that Clockwork is doing the work that I thought I was going to be doing, because we don't. Even though on the face of it we're both biology YouTubers, we actually work in concert with one another, because we have certain elements that make our channels unique. And if we can grow the hunger for deep dives into cell and molecular biology, which is something I say in every single video, it benefits everybody. Higher profile videos. If his videos get high up on the algorithm, it's good for me, vice versa, because there's like stem cells, infinite potential out there, and I want to see how far that potential goes. creating the cultural population of one's customer base. Exactly, like creating the demand for content like this. Because I think with big channels, there's no reason to risk you know, having content that is very easily accessible is not a risky move, right? It's very simple. No, I shouldn't say simple. It's straightforward to make content for a general audience. Like there's a known, well-treaded path of public science communicators that have treaded the path of general science communication to the public that has been worn. And that still serves an important entry point into deeper studies. And so Kyrgyzstan, I think, has that market on lock, and SciShow as well, right? Like the Green Brothers. And I actually do love that they exist. And I watched Kyrgyzstan and SciShow as well, right? I love that if they come, and from my YouTube analytics, I know they come, right? People watch SciShow and they watch Kyrgyzstan, and then they come watch me. I don't know if that relationship happens the other way around for those big channels, but I'm happy that I can exist comfortably in a niche that not many other people are in right now. Oh, I'm not going to say nobody, because, you know, I think Clockwork and I do share a niche. It's just not the exact same niche. And I hope, like, the aesthetics and the narrative of my channel also prevents other people from actually coming in and doing the exact same thing I'm doing. That would be actually crazy. If a new channel came on the scene and did exactly what I did, I would be shocked to think that someone else would do that. That would actually be, like, insane. Because, like, even other animation channels like Amoeba Sisters, we have very, very different styles. And I think it's neat to appreciate that. I missed a stream and I'll rewatch, but have you talked about the DNA binding proteins on sperm? I have not. So I kind of clickbaited everybody with fertilization because that was a convenient entry point into talking about what I really wanted to talk about, which is cell polarity, which is the idea of how to tell the head of an embryo from the butt of the embryo. That was the actual idea I wanted to talk about, but I think putting the title of cell polarity in the title wasn't going to, like, attract as many people, just because that's not an idea that you learn in Bio 101, I think. I mean, I didn't learn it until graduate school. So I wanted to start off with what people were familiar with and sort of work my way into talking about polarity. Yeah. Do you think science VTubers can eventually reach an audience as big as those channels? If you want to compare VTubers, you have to compare VTubers to VTubers. So Vedal, right? Vettel, the programmer who birthed Neurosama, the world's most successful AI-based VTuber, that's already kind of like a science VTuber, although what they do is mostly entertainment, but Vedal will do, like, coding streams. So there is proof that at the very highest level of Twitch, that kind of content can draw in people to watch. Whether or not a VTuber can get as big as Vedal just doing just science communication, we don't really know yet, right? That's why the arc of this channel is so interesting. The arc of vtheorem, I think, is one that I will fight tooth and nail to get as good of an idea of that as possible. So, like, if I'm talking about my ambitions for vtheorem, it is to see how far we can push this idea of there being a niche that is maybe underexplored. And that's very scientific too, right? Like, I feel more scientific joy right now as a member of vtheorem and trying to establish and define that kind of science VTuber niche as being my entry into innovation, we can call it, right? This idea that, like, underneath the bubbling surface of VTuber fans is this underserved, or people don't even know that they want it yet, kind of audience that we just have to strategically plan well enough to capture those people. Because part of why I think that there is a good chance that vtheorem could see more mainstream success in the future is because of reflecting upon my own sort of experience with streamers. I'm very sad to say that I no longer really watch streamers who only stream video games anymore. If they stream a game that I enjoy, I'll watch that, like Slay the Spire. Like, if you stream Slay the Spire as a VTuber, there's a chance that I will have already been in your stream at some point because it's one of my favorite games of all time. But I find that like the experience of watching people play video games for me has been, I just can't get into it anymore. And I don't really know why. I have some hypotheses as to why. I feel like every new game requires a little bit of buy-in from the audience. So if I haven't played the game, I don't really, there's less buy-in from me. But what I do watch streamers for now is just chatting. Like I think just chatting is one of my favorite categories of streaming, or people doing co-working and they're just yapping about their life or just telling stories and stuff, because I think that that kind of interaction, when it's not centered around a game which kind of competes for attention, um, is more enjoyable, more stimulating, you could call it. That's not to say that people who stream games are like making bad content. It's just like, for me, because the game is competing for the streamer for attention, I'm there for the streamer and not for the game, is what I'll say. So when people do things like draw and yap, I like that content as well. Just because I like it when the streamers are talking, and when they're not talking and it's the game, I get like taken out of the experience or something. And then the idea of listening to an expert talk about what they're passionate in is such a natural like next step for a just chatting stream, right? Like, so instead of just chatting about your life or whatever, just chatting about your work in the research that you've done, there's like niche corners of your interest, is like the next evolution of just chatting streams. And I think that like the pleasure of learning from experienced good science communicators, sharing in their joy when they are talking about what they're interested in, like when you hear science, when you the best case, the most I mean, everybody in vtheorem does this, right? When they talk about the thing that they're passionate about. But I think when I think of the visceral kind of like joy, is when Z is talking about space. It's like, it is so clear, if you've ever spent any amount of time on Z's stream when she's talking about space, it is extremely clear that she cannot hold in her enjoyment of the topic. And there's something very human and beautiful about that. Pureness is not the right term. The like unashamed, sincere embrace of a certain topic. And again, every vtheorem member has this, but I think the person that is the most genki of us, like the one that is outwardly expressing that joy, is Z. And I think that like that is why I think, there's potential for vtheorem, because it's like just chatting plus plus. At least I hope that we can convince an audience that it's just chatting plus plus. Where not only there's a format, right? There's a structure, there's a set list that you can expect, right? It's got reins on it, so the direction that a stream is heading in, and people talking about what they're into is always very, 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 very fun and interesting thing to listen to. And also, it just gives your brain something to play with, right? Like, Sidearray jokes about being like second monitor content, which I think is really funny. But it's also like second monitor content that gives you little treats to play with in your brain, right? So the benefits of a just chatting stream plus getting to unwrap this little piece of mental chocolate, I think that is what I would boil down vtheorem's appeal to. And I think that answering the question of do I think VTubers can get as big as Kyrgyzstan, VTubing itself is still kind of niche. But could vtheorem or could science, VTubers in general, get quite big or surprisingly big? I have to believe that is true, because science is driven by optimism, right? If I didn't truly wholeheartedly believe that there was a market for people who want just chatting plus mental chocolate, I would not have tried to make vtheorem happen, right? Like I have to believe that is true. I'll put it that way. So that was a long rant of me staring into the middle distance about reading chat. So let me catch up there. Have you read about the Mellangaster brain 3D map? Very fascinating indeed. I have not, but I should, because that sounds really cool. Clickbait is ruining a lot of things, sadly. So as a YouTuber, I will say this. Clickbait is actually a very interesting... Sorry. Clickbait, I think, is a very actually interesting communication tool. That's not to say that like I'm gonna clickbait everybody, but I think that what is objectively true is that if I were to name my titles without any element of clickbait, nobody would watch them, because and the way that I've kind of mentally worked around justifying having clickbait elements in a project is here. You have to meet people with where they are, and so you have to communicate in a way where it piques their interest. And of course, that's what clickbait is for. Now, clickbait turns into deception when you are not truthful about the contents of the stream. And I would say that this is the worst offender, is my stream here, where I talked about fertilization in the title, but in actuality, fertilization was only a small part of the story here. But that initial bit, like that deception, I've gotten over it in the sense where it's okay because people stay to learn the things that they did not know that they wanted to know, right? Because in order to communicate the things that I find cool, I have to make it clear to you that you should care. And clickbait helps me do that by establishing a connection between what I know and what a general audience might know. Now, the trick is once they read that clickbait is I wouldn't say I actually do clickbait, but I definitely think about clickbait when I think about thumbnails and titles. If I have their buy-in, then the job of the video is to capture their interest and keep them watching. And I think that's not unethical at all, right? Because like this stream, like Cell Polarity, if I had titled the stream Cell Polarity, I don't think people would care, because cell polarity is not a commonly understood concept in biology, and B, you don't have the background to even know why cell polarity is cool. So instead, I clickbait with fertilization and a sperm and an egg, just for a little bit of fun, and so that you show up so I can actually talk about what I want to talk about. Let me yeah, that's how I would conceptualize clickbait. It's selective, it's nuanced, it's interesting as a tool. It's not all bad, is what I'll say. Yeah. I love listening to Phi talking about his work and simultaneously reading fun facts about fertilization. Yeah, exactly. And I'm glad that I'm providing that kind of entertainment, because I just think it's a little more fun to listen to than the typical Just Chatting stream. But there's nothing wrong. I still show up to Just Chatting streams too, by the way. Like, there's nothing wrong with that. But when I want something for my brain to play with instead of just chatting, I want to see more people talk about expertise. Yeah, again, it's just like mental chocolate, and that's not. I've talked about this before about elitism. I don't think that everybody has to be like high-minded about the content they consume, right? Like, I watch JoJo Bizarre's Adventure, and it's one of my favorite animes of all time. It's not that intellectually challenging, right? Your media doesn't have to be intellectually challenging, but when you're in the mood for something that tastes good to your brain, it would be nice to have a niche that you could go to have that kind of brain candy satisfied without maybe the buy-in from a video, right? So that's why I do video and streams, because videos, that's my like very carefully crafted, I had a lot of time to think about how to say things. But you might not necessarily want to put on a video on your second monitor. You might want the lower buy-in of a yapping stream with elements of that mental chocolate. Like maybe you're in the mood for a couple of chocolates and not in the mood for a whole like Easter bunny level of chocolate consumption, which is what a video is. But sometimes you are in the mood for a video, and so that's why I want to do both elements. Yeah. Yeah. It's important to get one's foot in the door. Exactly. Clickbait, it can be bad, but it is important in the pipeline of videos. I mean, I would have cared, but I also have a degree in bioengineering with a minor in biomedical engineering. Exactly. Like, I have to be mindful of two things. I have to be mindful of the fact that I have courted an audience that is more deeply invested in biology, but also keeping in mind that there are a lot of viewers, and I'm extremely like happy that I have viewers that don't have biology background who repeatedly show up to these streams, by the way. That is a very good indication that I am on the right trajectory in terms of where I want to be for the future of this channel. And so keeping all of your interests in mind is kind of where I want to live. And it's actually the perfect place for me to want to live, because while I love teaching in like a university environment, I always have to be mindful of like, I need to talk about the things that are going to get them jobs, because ultimately in the current year, that's what university is for. Whereas if it's for an entertainment audience, then I can talk about whatever, as long as you trust me to make things cool. And I will treat your trust with the utmost respect by, working as hard as I do on making these presentations. So I think it's a fun symbiosis, I guess we could say. Welcome, AG. So yeah, we're at the end and I'm just yapping right now. So we did talk about cell polarity, the study of understanding how cells understand head from tail. But now we're just kind of yapping, which I will end soonish as I get ready to eat lunch. By the way, happy birthday, happy Valentine's Day. Could be my birthday. I die every 5 to 8 days, so it could be my birthday. Yeah, happy Valentine's Day. It's good. It's good. I hope you all go out and buy some chocolate. I guess I'm thinking about mental chocolate because of chocolate discount day, which you should absolutely all get. Yeah. Your presentation is always a blast to attend. Thank you so much. Thank you so much. I really owe a lot of the design and style of all of this to my previous mentors and teachers in graduate school, because when they gave me mental chocolate by doing the presentations, how they do it, it made me understand how to provide mental chocolate for you all. So it's good. Chocolate discount day, where well, in the US at least. How does one calculate a birthday if you don't live a full year? I just celebrate every day that I spawn, I guess, or every day that I differentiate. Yeah. Okay, so last things to mention. Pinned in the comments in the live chat is a link to the Small VTuber Awards, where you can nominate vtheorem for all sorts of different categories. I will do the thing. Is it awards or award? There we go. If you want to help out vtheorem in terms of getting us a little bit more prominence, you can help us all out by voting for us for these particular categories if you have that little bit of time. And if you join the Discord, there's actually a post in the announcement channel that makes it even easier for you by providing the links to vtheorem socials. So I'll go ahead and plug that Discord one more time. Oh, status on the Sigma chocolate. It's so their giveaway ends on the 20th. So on the 20th, I will know as to whether or not Sigma Aldrich has given me chocolate. If you didn't know, I've been on a sort of quest to get Sigma Aldrich, Millipore Sigma, to send me their very famous lab chocolate. They sell these adorable bottles that look like the chemical bottles that you would get for your reagents, and instead of, sodium chloride or whatever, it's a Swiss chocolates inside, and they're very, very cute, and apparently they're very, very good. And I so badly want to buy, or not to buy, I want them to give them to me. I want to earn them the way that they decide to give me the chocolates. I think Leo in the Discord had mentioned that because they don't have a trademark on the chocolate, you could just make your own. But it's about the symbolism. It's about the romanticism of Valentine's Day. I want to be courted by Sigma. So I've been tweeting at them for the past 2 weeks, begging for the chocolate. So my Twitter is in the channel description, the description for the stream, if you want to go check out how that's going. But yeah, so I entered officially their giveaway when they opened it on Valentine's Day, and I am patiently waiting with my hands interlocked, just waiting for a DM from my beloved Sigma Aldrich for that good old lab chocolate. Okay, with that being said, thank you all for attending a very special Valentine's Day stream. I have no idea what we're going to be doing in the future. I am working pretty hard on getting this flu video out. It's probably going to take me another month. Hopefully people will still care about the flu by then. But also, before that, hopefully people still care about the flu for my video statistics, but also hopefully people don't care about the flu because it didn't become a huge freaking deal. So, you know, fingers crossed for that. With that being said, it was nice to get to chat with all of you again. Thank you for attending. I hope you learned something cool today about how cells understand their heads from their tails. And I will see you next time. Bye-bye, y'all. Bye-bye.